Hello, hello. Today we're going to be talking about glycolysis, gluconeogenesis, glycogen, a lot of stuff with sugar, and a whole bunch of stuff on vitamins. Actually, not that much housekeeping. I'm not a TA anymore. I guess the only thing is, so again, I made these lectures last year, and then I sort of edited them. So yeah, a lot of maybe some of the fat from REM's lectures is cut out. It's much more NBME directed. I actually added a lot of features, but I think this lecture is much more aligned with what you'll see in first aid and what you can expect to see on the NBME. Some of the stuff, it's really difficult to organize these lectures so that you don't at all reference things that are going to appear in future lectures, like the vitamins, a lot of the vitamins, like the methylation cycle. It's hard to introduce the vitamin without talking about the methylation cycle. But trust me, all of this is stuff you will have to know for MFM. So better learn it, now than later, I suppose. Okay, today we're going to be talking about just general overview of carbohydrates, talking about glycolysis, PDH, acetyl-CoA, the Krebs cycle, electron transport chain, and ATP synthase. What's the purpose of lactate? A huge section on vitamins, which is really going to get into a couple of things we'll see in future lectures. Making new sugar in times of need when the body's hungry. Glycogen, building it up, breaking it down. The pentose phosphate pathway, also known as the HMP shunt. Just general regulation. I want this to be intuitive. I want this to be a logic. And a couple of metabolic disorders, mostly focusing on glycogen disorders and a couple of poisons. Okay, carb overview. Okay, glycogen is only going to be in the liver and in the skeletal muscle. The liver, I'll say it again and again, is altruistic. It's taking this glycogen and it's going to altruistically break it down and feed the rest of the hungry body. The skeletal muscle is more selfish. It's like, Smog the dragon sort of hoarding its gold. Fats can be broken down into glycerol, which can enter into this pathway. So can proteins and muscle be broken down. Amino acids can enter this pathway. Typically, we're going from glucose to pyruvate. That's pretty much all cells. Some cells, really mostly just the liver that we need to focus on, can go backwards. So the liver can do gluconeogenesis, making from pyruvate and glycerol and amino acids. It can actually go, it's kind of the reverse of glycolysis. There's no oxygen, or we don't have a mitochondria like we'll see in the red blood cells. We're going to go ahead and make lactic acid. But most cells, though, we're going to take our, pyruvate, convert it to acetyl-CoA, put that in the Krebs cycle. What is the point of all of this? It is to make NADH and FADH2. These are like casino chips. A casino chip, you could have a casino chip for $10. You can't take that to the corner store and buy something with it, but it's effectively $10. You just need to, there's a very specific, mechanistic way that you could trade in that casino chip to get actual currency, which is taken everywhere. That's our ATP. So how do we do this? How do we cash it in? We take it to the electron transport chain. We put it into chemiosmosis, which really just means, the protons flowing through ATP synthase, and that generates ATP. Another more broad overview, glycolysis happens in the cytosol. We take one glucose, it's 6 carbons, we turn it into 2 pyruvates. These are 3 carbons. This makes a teeny little bit of ATP. We call this substrate level phosphorylation. We also make a little bit of NADH, which is more exciting, these casino chips. The pyruvate, each one of them goes into the mitochondria, specifically the matrix. Here we see the matrix. Here we see the inner mitochondrial space between, the outside membrane and the inside membrane. So in the matrix, this pyruvates turn into acetyl-CoA. This generates a little bit of ATP. This generates NADH. This acetyl-CoA then goes into the Krebs cycle. Here we make a little bit more ATP. And then just notice we have, this is for the birds, right? We have a tiny amount of ATP in glycolysis and Krebs cycle. Mostly what we're interested in is these casino chips, right? We get a little bit in the first part, a little bit over here, and a little bit over here. NADH, FADH2. NADH is 2.5 ATPs is how much it's worth. FADH2 is 1.5. Now that might vary depending on like different shuttling mechanisms, but it's really not that important. So we're going to cache these in again, the electron transport chain, and that's actually where we're getting most of it. This is oxidative phosphorylation because it involves oxygen. Oxygen is the final electron acceptor. Glycolysis. So terminology, kinases are going to involve ATP, whether we're creating or destroying. They're going to be adding or taking off a phosphate from a protein or a molecule. A phosphatase specifically is going to take off a phosphate, but it does it via hydrolysis, right? It doesn't transfer it to an ADP. You are going to use an organic phosphate to you're going to use water to break off this phosphate. So you generate an inorganic phosphate. A phosphorylase does exactly what I was just saying. It takes a phosphate and actually uses it as a nucleophile to break something apart. We're going to see this mostly in glycogen breakdown. And then a dehydrogenase. So we saw that a kinase could make ATP or lose ATP. Dehydrogenases, I'm pretty sure, I can't think of any examples. We're really only going to be making NADH and FADH2 and NADPH. So kinase, phosphatase, phosphorylase, and dehydrogenase. Here we see a good picture of glycolysis. I would very much note the carbons and when we're generating ATP and NADPH and when we're breaking it in half. So might you have to know every single molecule and every single enzyme? Yeah, it's fair game. I think it's lower yield. I would definitely know when you see a kinase that involves ATP, when you see a dehydrogenase that involves the generation of NADH. One instance I can think of where it makes an FADH2. Probably the more important thing is to know the first half, which is the investment phase, and the second half, which is the payoff phase. What separates these two halves? We're going to cut our six-membered hydrocarbon into two separate three-carbon structures. So anytime you see a kinase in the investment phase, well, that's involving an ATP because it's a kinase. And it's the investment phase where we're making an ATP or losing an ATP. You are losing one ATP there. You are losing a second ATP there. So this is minus 2 ATP in the investment phase. In the payoff phase, I'm seeing a kinase, but it's the payoff phase. So am I making or losing? I'm definitely making. We see another kinase, so I'm making 2 ATP. So here there's 2 ATP and here there's 2 ATP. But remember, we split the molecule into 2 3-carbon structures. So we're actually doing the payoff phase twice. So you make 4 on the payoff, you lose 2 in the investment, so your net is 2 ATP. I also see a dehydrogenase, and that is also times 2 because, again, you're doing the second part twice. So you're making 2 NADHs and 2 ATPs. You can also kind of reason your way around what the enzymes are because they're often but not always named after the reactants. It's like a kind of loose rule of thumb. Glycerol can enter into glycolysis, can become DHAP, dihydroxyacetone phosphate, which can become G3P. Definitely, you should be familiar with the structure of glycerol. So this is our glycerol, and then this is our fatty acid. And we have 3 of them, so the whole thing is a triacyl glycerol, triglyceride. 1,3-BPG, which we see here, and red blood cells can actually be converted into 2,3-BPG, which is going to bind onto hemoglobin, stabilizing the TENS conformation, which really just means that we have a lower affinity for oxygen. This would be useful, if you're, a Sherpa up in the mountains and there's a low oxygen environment, you actually might have higher levels of 2,3-BPG than most other people. All of the enzymes of glycolysis are reversible, except for 1, 3, and 10. Now, why do I say 9, /10? It's because some people consider, they don't really consider this a step, so they might say 1, 2, 3, 4, 5, 6, 7, 8, 9, but I'm just going to call it 10. But just if you see it mentioned as 9, that's probably why. Hexokinase, irreversible. Phosphofructokinase, also known as PFK1, irreversible. And pyruvate kinase, irreversible. All of these are kinases. In the investment phase, we are losing an ATP, and in the payoff phase, we are making an ATP. Because they're irreversible as well, they're going to be highly regulated. The regulation mostly is just working at negative feedback principles, right? If your body gets hot, your brain's going to go ahead and start making you sweat. That's going to cool you down. Once you're cooled down, this feeds back into the system to tell your body, hey, we can stop sweating because we are actually a good temperature now, right? So everyone should be very familiar with negative feedback. Everything is going to operate on that. Hexokinase is this first step. What is the significance here of this? It's a trap. So glucose is going to enter cells through a GLUT, G-L-U-T, glucose transporter. It's transmembrane protein that lets glucose in. Now, we want glucose to get into most of our body tissues and not get out, right? Because I want it to get into my muscles or my fat cells and stay there so my body can use it. That's the whole point. Well, why would you put it in and take it out? There is one exception to this, which we'll get to. And so this phosphate traps it in because once it has this negative phosphate on it, the glucose transporter will not let it through. It will not let it leave the cell once it has this. So this is trapping the sugar in, which is definitely important. And it operates off of negative feedback. So the glucose gets in through the glute. It's right here. It could go out, but if it interacts here, now it cannot get out. It's not allowed out anymore. And once we have enough of this, it's going to feed back and inhibit the hexokinase enzyme. So hexokinase versus glucokinase in the liver cells is glucokinase. So why would a normal body tissue take in sugar? Because it's hungry. It's hungry and needs energy, right? And so if you have like a low amount of body sugar, I still need my brain to take in sugar, right? My brain always needs sugar, even if I have a low blood sugar. So I want it to have like a very high affinity. It'll easily and readily grab onto sugar in the blood and push it into the brain cells. That's what most tissues do. But the liver is different. The liver is very wealthy, right? It has tons of energy stores. So the liver's reason for taking sugar out of the blood is almost never that it's hungry. It's actually the opposite. There's too much sugar that it's dangerous, right? You just had 8 brownies and your sugar levels are skyrocketing and your liver goes, holy crap, there's way too much friggin' sugar. We need to take some of this out. So we need a system then based on that concept where the liver glucose transporter and the first enzyme actually has a really low affinity. It's a really low affinity, which is to say it's a very high Km, so that only when our blood sugar levels are really, really dangerously high that the liver goes, ugh, all right, fine, I'll save the day, I'll take this out, and I'll build it into glycogen or I'll build it into fat or something. Additionally, a normal body tissue, like a brain cell, like, yeah, sure, we want it to like really easily grab onto sugar so that it can, make ATP and live. But there's like a finite amount of sugar that any normal cell typically needs. So we're going to put like a maximum amount that a hexokinase could interact with, because there's a fixed amount of sugar that we need to get into the brain cell. But again, the liver is different, right? If I eat 3 brownies, okay, well, that's too much sugar. My liver needs to take over and grab some of the sugar out of the bloodstream and put it in glycogen or fat. But what if I have 8 brownies or 15? There's no limit to the amount of brownies I could eat. And my blood sugar could be absurdly high. And so we want a Vmax to reflect that. We don't want the glucokinase in the liver cells to tire out. So the Vmax is really high here. PFK1 is probably the highest, most highly regulated enzyme of glycolysis. Note a lot of these things are really just going to be negative feedback. The whole point of this is to make ATP. So if I have a lot of ATP, that is going to negatively feedback. Citrate is something we see in the Krebs cycle. If I'm making a lot of citrate, then that's also going to negatively feedback. And if I have low ADP and AMP, well, this is going to be indicative of a low energy state. So this is actually going to be positively regulating this enzyme. So, yep. And then pyruvate kinase, the last enzyme of glycolysis before we put it into the mitochondria. AMP indicative of a low energy state. This is telling us we've got a big shipment of a whole bunch of stuff beforehand, so we need to take it in, and there's a bunch of stuff coming your way. So go ahead and start working really hard so that we don't. It's kind of like if you go to Chipotle and they see a huge line, so that they start speeding up, right? Even the person who's the last in the assembly line. And ATP, negative feedback, also means that you have a high energy state. Acetyl-CoA, this is the product, this is negative feedback. Alanine's kind of weird, but this is just going to be a sign that building blocks are abundant. So it's similar, it correlates with high ATP levels. What's the point of all of this? I mean, you guys kind of know, but I remember the first time I learned glycolysis and Krebs cycle, it all seems so weird. Like, are we trying to make energy? What is the point of this? Yes, we are trying to make ATP. So to make ATP, we take ADP and a phosphate, and we smack them together. But this is a very endergonic process. It requires a lot of free energy to occur. And we're going to get that energy by slowly and surely oxidizing glucose. And we mentioned the casino chips. We have NAD+, also known as B3, also known as niacin. This is 2.5 ATP. I remember that 2.5 rounds up to 3, so it's B3. There is an oxidized version and a reduced version. Notice that we have here's 1 hydrogen here and an extra hydrogen here. So you might remember oil rig from Gen Chem, Orgo, and Biochem. Oxidation is loss, reduction is gain. We're mostly talking about electrons, but we still are, right? But when we're in biochem, usually the electrons are hidden in the form of a hydride. Now, that is distinguished from a hydrogen or even a proton. This is an electron-rich hydrogen source. So there are extra, hydrogens there. Even notice, look at the charge, NAD+ to NAD H, right? We became more negative from a positive to a neutral. That's because there are electrons in this hydrogen. That is to be distinguished with something like OH minus to become H2. Oh, what happened here? We gained an extra hydrogen, we became more positive. Here we gained a proton. Here we gained a hydride. You don't have to get too into the weeds with that, but it's just like a useful distinction, I think. The name itself, NAD, nicotinamide adenine dinucleotide. Well, this is a nucleotide, right? We've got a ribose sugar, we've got a phosphate, and this is technically nitrogen base. And then the same thing here. We've got this, and our adenine. So nicotinamide is one of the nucleotides, and the adenine is the other, and there's an oxidized version and a reduced version. Similarly, we have riboflavin, which is the component that makes up FADH2, and this is also in the name. Flavin, which is this nitrogen base. Adenine, which is this nitrogen base. Dinucleotide. Well, this is, here's our, nitrogen base. Here's our ribose. Here's our phosphate. This one looks weird, but this is still a phosphate, and this is actually also going to be a ribose. It's just a linear one, and this is also a nitrogen base. So you can kind of see the whole thing right there. So that's one nucleotide, and that's the other nucleotide. And similarly, there's an oxidized version and a reduced version. We are really just gaining a hydride, so nothing here, something there. And this is 1.5 ATP, and I just remember that 1.5 ATP rounds up to 2, and so that's how I know that FADH2, the riboflavin, is associated with B2. Once pyruvate's made, it's going to go into the mitochondria in the matrix, and it's going to be converted into acetyl-OA. Now, the SparkNotes version of this is that we attach this CoA group on, which is actually a large functional group. We get out of CO2, so 3 carbons becomes 2 carbons. We generate an NADH as well. So that's going to be 2.5 ATPs stored as a casino chip. Now, the PDH is actually a kind of complicated enzyme. There's really 3 different domains we need to think about. There's the decarboxylase to cut off one of those carbons. There's a transacetylase to put on the CoA SH group, so that thioester group. And then the dehydrogenase just regenerates the E2 because it's kind of tired after it does its thing so that it can, interact with another pyruvate. There's a lot of cofactors you need to know. I would remember tender, loving care for Nancy. T, thymine, L, lipoic acid, C, CoA, SH, F, FAD, and then N, NAD+. These are all things that we need to make this work. Acetyl-CoA really just breaks down into a phosphorylated ADP, pantothenic acid, which is vitamin B5, and I remember panto is kind of like pento, which is 5, and then an acetyl group. And the acetyl group as well, I would recall, is really just the that we had the pyruvate was 3 carbons, and then we cut off one, and then these are the 2 carbons that are left over, and so this was the CoA that we attached to it. Super highly regulated PDH, maybe the most highly regulated enzyme of metabolism. I would know that if you put a phosphate on it with a kinase, that's going to turn it off, and if you use a phosphatase to take off a phosphate, that's going to turn it on. So this is like a catabolic enzyme, and I would kind of recall that this is like a general pattern. We'll get much more in detail into this later, that for catabolic enzymes, having a phosphate means they're off and no phosphate means they're on. So now we're in the mitochondria, as we said, so let's take a deeper look at the Krebs cycle. So acetyl-CoA is going to combine with oxaloacetate. So OAA is oxaloacetate. Why is this called a tricarboxylic acid? Why is it called a Krebs? I don't know, some science guy named Krebs, but we have these 3 carboxylate groups for a lot of these structures, especially citrate, hence the name. So 4 carbons for oxaloacetate combined with 2 carbons for acetyl-CoA is going to make us a citrate. We're going to isomerize that to isocitrate. Now use isocitrate dehydrogenase to make alpha-ketoglutarate. Notice that the dehydrogenase is here, so absolutely we're making NADH, and we're splitting off one of our CO2s. So it was 6 carbons, and now it's going to be 5 carbons. And right after that, we have another dehydrogenase, so we're making another NADH. And again, like whenever you're not sure, just remember that things are often, but not always, named after their precursors, right? Isocitrate dehydrogenase, the precursor is isocitrate. Alpha ketoglutarate dehydrogenase, the precursor is alpha ketoglutarate. Here, note that we're also attaching an S-CoA onto the molecule. And so this is going to be very much like the PDH multimeric enzyme, and it needs all of the same stuff. Tender loving care for Nancy. Thiamine, lipoic acid, acetyl-CoA, FAD, and NAD+. Succinyl-CoA synthetase makes a GTP, but GTP is effectively ATP. That's the same thing. Then succinate dehydrogenase. Again, we have another dehydrogenase. This makes FADH2, though, which is worth less. This is 1.5 instead of 2. This is weird because it also happens at complex 2 of the electron transport chain. So it's just kind of coupled. Rather than making FADH2 that then floats over, it makes it at the complex itself. Fumarase makes malate, and then malate dehydrogenase, another dehydrogenase, and ADH makes oxaloacetate. So just imagine that when you start this, you could just have one oxaloacetate. You could have a billion acetyl-CoAs, but one oxaloacetate, and you'd be fine. It would keep running, right? Because one and, this is 100, and now we have one of these, and then one of these, and one, and it just keeps converting one. And we're losing the 2 carbons that we put in every time, this carbon and this carbon. And now the number is still 1 at the end of this. We're regenerating everything, but this is now 99. So this is going to be important when we get to ketogenic versus glucogenic amino acids. I know that's a lot, and I went pretty quickly. So please, can I keep selling seashells for money, officer? Pyruvate, citrate, isocitrate, ketoglutarate alpha, succinyl-CoA, succinate, fumarate, malate, oxaloacetate. Technically, there is it's pyruvate acetyl-CoA. So sometimes I think of like, please, can I keep selling seashells for money, officer? Make sure to count the carbons, right? This is 2. This is 4. 2 plus 4 is 6. We isomerize to isocitrate. Alpha ketoglutarate knocks off the first one. Isocitrate dehydrogenase knocks off the first one. Alpha ketoglutarate knocks off the second one. And now we're good. Now we're just 4 carbons, and it's 4 carbons the rest of the way through. Anytime you see an dehydrogenase, you're making an NADH, except the one time they make an FADH2. So just if they showed you a structure, I think it's really low yield to know all of the structures, or at least to go out of your way to memorize them. You can kind of just make intuitive reason around this. So remember that in biochem, oxidation is loss, reduction is gain, and we're really just looking at hydride. So that's going to be electron-rich hydrogens, typically on carbons. So here, ignoring the hydrogens on oxygens, this is C6H12. So it's kind of like a 2-to-1 ratio. So we would say that this is highly reduced. There's a lot of energy in this glucose. Now we get the pyruvate at C3H3. It's been significantly oxidized. There's a lot less energy, but there's still some. And then we're going to go to malate, which is C4H3. And after that, we're going to go to oxaloacetate, which is the most oxidized. And we've squeezed all of the juice out of this lemon or whatever, right? So that if they show you, if they give you these 2 structures and they say which one is which, one, if they tell you one, it'll never be this pointed, but if somebody gave you this structure and this structure and said one of these is malate, one of these is oxaloacetate, you should be able to count that and know which one is which. Again, I don't think this is a high yield, but it's potentially useful. Okay, so this is kind of the moment we've all been waiting for. This is the big showdown. What is the point of all of this? Will it pay off? It's kind of like you give somebody a bunch of money and he's like, I got a surefire way to make billions. And it seems like complete silliness until the last minute. He comes through. So that's the electron transport chain, and ATP synthase is everything kind of coming together. Why do this whole thing, right? If glucose is so energy rich, why don't we just oxidize it and immediately take that energy and put it, into whatever we need? Well, first of all, it'd be like if you oxidize glucose all at once, that would be like saying, why don't we just use up all of the energy in your car all at once to get you from, your home to Stony Brook Med or whatever. It'd explode, right? You can't just have a full oxidation of everything all at once, or that's too much energy. We want to eke out a little bit of energy here, a little bit of energy there, and things are much more regulated too when you do it this way. So there's a whole fun evolutionary argument for why it works in such a complex fashion, but that's definitely not going to be on the test. But maybe important for the electron transport chain just to know that we're slowly but surely, eking out little bits of energy as we pass the electrons from proteins who want them less to proteins who want them a little bit more, ultimately to oxygen, the final electron acceptor, who wants it the most, which generates water. Let's not forget where we are. We're in the mitochondrial matrix. Here's the outer membrane, here's the inner membrane, and the electron transport chain is in the inner membrane. So, here we're going to have all of our complex, 1, 2, 3, 4, and the ATP synthase. And we're pumping protons from the matrix into the intermembrane space. That's the whole point. And so this is going to be a very low pH here, and which would mean it's a high hydrogen or proton concentration. So this is maybe like a good picture. Ignore vitamin K right there. So complex 1 gets all of its energy from electrons. That's the electrons from NADH. Remember, that's 2.5 ATP at the end of the day, right? It's not going to be such a clear exchange, but just if you had 100 NADHs, you're going to get about 250 ATP. So it goes from complex 1 to coenzyme Q, also known as ubiquinone. This goes to complex 3, which goes to cytochrome C. Remember that if cytochrome C gets out of the mitochondria, it causes apoptosis. Goes to complex 4 and ultimately goes to oxygen to make a water. Complex 2 is the FADH2. Remember, this is the succinate dehydrogenase. It happens directly here, succinate to fumarate. That goes to complex 2, and that goes to 3 to cytochrome C to 4. So it goes 1 to Q to 3 to C to 4 to oxygen, or it goes 2 to Q to 3 to C to 4 to oxygen. Easy, right? Easy as pie. And what's the whole point of this is when we give the electrons, we're giving the electrons to things that want them more than the previous thing. And if I don't want an electron that bad and I give it to somebody who wants it a little bit more, that's exergonic, just by a little bit, right? I'm giving a little bit of free energy. And so what do the proteins do in this intermembrane with this energy is they take protons that are sitting here and they just kind of lift them. They throw them way up high where they don't want to be, right? And it takes energy to put things where they don't want to be. We have this is very positive. So, and there's a lot of hydrogen. So we're pushing it against its electrochemical gradient, these protons, and that requires energy. And that energy comes from the electrons from NADH and FADH2. Whew. Okay. And then at the end of the day, we got all of these protons here. And so the protons are going to flow in through the ATP synthase. This is known as chemiosmosis. And this is just more energy. The whole thing's weird, I know. This is just more energy that we are, what are we doing with this energy? We're now taking ADP and phosphate, which we said this is very endergonic, coming together, and the energy comes from the flow of these protons. That's if everything works perfectly. Often these electrons go awry and they'll interact with oxygen to make superoxide. But this superoxide can be dealt with, right? We have superoxide dismutase, which can convert it to hydrogen peroxide, and catalase and glutathione peroxidase can make water, which is, of course, quite benign. So I actually would know those enzymes: superoxide, catalase, and glutathione, and then the superoxide, hydrogen peroxide to water. Just another fun weird fact, like we just said, succinate to fumarate, succinate dehydrogenase happens literally at complex 2. And of course, that's our FADH2, which is only 1.5 ATP, which runs up to 2, and that's why it's vitamin B2. There's also a lot of poisons, which might be low yield to know. Rhodinin and metformin. Metformin is a type 2 diabetes drug, inhibit complex 1. Antimycin can do complex 3. Cyanide famously does complex 4. And then oligomycin can do complex 5, which is really just the ATP synthase. So we said this, but maybe the best way to conceptualize this: protons flowing down the ATP synthase, kind of like water going downstream, right? Like the water, the gravitational potential energy to push the water from up high to down low, right? It happens automatically. Similarly, if there's this is very positive and there's a ton of hydrogens, there's a double incentive, an electrical incentive and a concentration incentive for them to flow here. And when it does, some of that gain in entropy is somehow captured by this complex 5, this giant ATP synthase protein, in the same way that this water wheel captures the energy from the water and can use it to churn and power something within the, this factory, this building here. Similarly, we're going to capture this energy and use it to combine these things to make ATP, which is now a little stored unit of energy that's, like the euro. It's good everywhere in Europe, and we can spend it wherever we want. So on that note, mitochondrial uncoupling. This happens with a drug called 2,4-DNP. So let's just recap. The protons flowing down the electrochemical gradient from the inner membrane space to the matrix is going to be captured here as ATP. But that's only because it's like meeting resistance, the same way that the water goes, across the water wheel and turns it. But what if we're cheating the system with this proton leak? This proton leak is the 2,4-DNP. It's shuttling the protons from the inner membrane space to the matrix. It's cheating the ATPs. We worked really hard to get these protons concentrated in the inner membrane space, specifically and only so that they could flow down ATP synthase. If we're shuttling them then across for free and not capturing any of this energy in ATP, then this sort of defeats the whole purpose. A lot of people use this as a weight loss drug until it was, it worked too well and people started dying, right? And that's because you're cheating your body out of. You could eat a lot of food and not get that much energy from it, is the idea. I think Russian soldiers also used it during like World War II, because if you're not capturing the energy and chemical energy as ATP, then the energy's got to go somewhere, and it just becomes heat. So it would heat people up. Summary so far: this is in the cytosol. 1 glucose becomes 2 3-carbon pyruvates. We make 2 ATP, 2 NADHs. There's an investment phase and a payoff phase separated by the point where we break it in half. This then becomes acetyl-CoA, which generates an NADH. It loses a carbon, so now we have a 2-carbon acetyl-CoA. This goes in the Krebs cycle, combines with oxaloacetate to make a 6-carbon citrate. We isomerize, then we do isocitrate dehydrogenase, pop off a carbon. We have 1 carbon left from the original structure. Of course, we're doing Krebs cycle twice because we get 2 pyruvates, but after we have, the alpha-ketoglutarate, now alpha-ketoglutarate dehydrogenase, named after the reactant often, and dehydrogenase tells us there's an NADH, is going to pop off the last CO2, and blah, blah, blah. We're making lots of NADH, lots of FADH2. This is substrate level phosphorylation, which really just is anything that's not oxidative phosphorylation. Haven't made much. We made 2 over here, and we made 2 over here. Remember that the GTP here are really just ATP. The whole point of this is to make these casino chips that we cash in at the electron transport chain. We pass them from proteins who want them less to proteins who want them more. And every time we do one of those little exchanges, that is a favorable exergonic thing. And what is the exergonic means? We're releasing a little bit of energy. How is that energy used by the protein? It's used to pump protons from the matrix into the inner membrane space. And when they go into the inner membrane space, there now is a very high electrochemical incentive to flow back down here, but they're typically not allowed to just go anywhere through the membrane. They're protons. They can't go through the membrane unless you're a Russian and you're taking 2,4-DNP. You have to go through ATP synthase, which is this water wheel type thing. And the protons flowing down that is like water going down a mountain, which sort of rotates and churns the ATP synthase complex 5 so that it can combine ADP and phosphate to make ATP. This is called oxidative phosphorylation because the last electron acceptor during the electron transport chain is from complex 4 to oxygen. Oxygen is, hence the name oxidative. So keeping score, 2 ATP, 2 NADH, this is 2.5, gives us 7. Here we, everything we're doing now is going to be twice. We get 2 pyruvate. So we're, and again, remember that this 2 here was a net. We put in 2 and we spent 4. We put in 2 and we got 4. Pyruvate now we're making 2 NADHs because we're doing it 2 times, so that's going to be 5 ATP. And in the Krebs cycles, we're making 3 NADHs, 1 FADH2, and then 1 GTP, which is really just an ATP, but we're doing that 2 times as well because we have 2 acetyl-CoA, and that is going to be 20 ATP. Now, this will vary a little bit depending on the cell type and the shuttling mechanism, but it's about 32 ATP that you're getting. Lactate dehydrogenase fermentation. So pyruvate is very versatile, as we'll see. It can become lactate. This generates an NAD+. So this is very important. This is NADH to NAD+. Usually we see NAD+ to NADH. This is weird. We'll come back to that. When does fermentation occur? If you have oxygen, you're typically going to be oxidative phosphorylation. If you're fermenting, or really just fermentation here is, it's like a red blood cell doesn't have a mitochondria, so it cannot do any of the cellular respiration stuff. So it has to do anaerobic respiration, which is fermentation. So red blood cells, or if your muscles are working really, really, really hard, There's maybe, more demand than the oxygen can meet up with. And so some of your muscles are going to be doing fermentation, and you probably had a gym or PE coach say, you got too much lactic acid because, maybe you're pumping too much iron and you run really quick and the lactic acid makes you sore. And in yeast, this makes ethanol, and this is often how we get things like beer, but we're making lactic acid, also known as lactate. So that's basically when if you're a red blood cell, there's too much of a demand, not enough oxygen. The question now is, why would you do fermentation? If we look at this, right, this is a glucose to pyruvate. We're making 2 ATP and we're getting 2 NADHs. But the whole point of the NADH is a casino chip that we cash in at the electron transport chain. And of course, that gets us ATP, but it also regenerates our NAD+, which can come back here. What if we couldn't do that? Well, then what's going to happen? We're going to continually make all of these casino chips, all of these NADH that we can never cash in, and we're actually going to run out of the NAD+. You need to put in NAD+ for glycolysis to occur. So if you did this forever and ever and ever, you would run out of NAD+. Glycolysis would stop, your cell would die if you cannot regenerate with the electron transport chain. So if you can't do that, well, then you make lactate. And this regenerates the NAD+ so that you can just continually feed it into glycolysis. Vitamins. Okay, so this section is a doozy. There's a lot of info here. This is probably what I changed more than anything else compared to last year's lectures. All right, so vitamins really just defined as anything that's essential for growth, but which we can't make internally, and so we have to synthesize it. Here's a lot of the vitamins you might be familiar with, and of course, many people take pills and supplement their diet that way. ADEK, ADEK, these are all the fat-soluble ones. I would definitely know that. So they can just move right through a membrane like the non-polar hormones. B9 and B12, these are associated with a lot of with blood and with myelin and neural tube issues. Vitamin C and E are for antioxidation. And then these play a variety of role, but these are really in the weeds with a bunch of metabolic processes. But we're going to go through all of these in crazy amounts of detail, but I think it's important. So let's take a look. So it's like, so, this is kind of like saying, what's the best religion or political party, right? What's considered a healthy food? Not a debate I'm looking forward to, or that I could even really speak intelligently about. But the idea is that if you eat like meats and chicken and you're eating a broccoli leaf every now and then, or a carrot or a potato, you're just eating like a mildly decent diet, you're probably going to get most of the vitamins that you need. You have an apple every once in a while. You have a couple of peanuts or almonds or something. So now, of course, there's a couple caveats there, but that's what you need to know for the NBME step one. As long as you have a decent diet and you're not just eating potato chips all day, you will probably be good for a lot of your things. Now, there are a couple, so where is vitamin B2? I don't know. Just assume it's in a bunch of like nuts and oils and fish and broccoli. I wouldn't overthink it, and I wouldn't stress too much about it. Exceptions you should know: B9. This is folate. I think of foliage. This is green leafy vegetables. You need to know that. B12. Remember we said we haven't said it yet. Never mind now. I'm in my own head. Cobalamin, which we'll talk about, is like Kobe beef. So I always thought of it as like, this is the meat, dairy, eggs. This is the one that people who are vegetarian, vegan have issue with, and so they have to often take B12 supplements, or there'll be tons of blogs about where to get the most B12 if you're a vegan. Vitamin C is in fruits and veggies. Vitamin D is in meat, dairies, and eggs, kind of like cobalamin, but we can also get it from the sun. The sun's going to cholesterol into vitamin D. And K is also green leafy vegetables like B9, but importantly, those are the precursors in your GI. The gut bacteria are going to transform those precursors into the finished vitamin K product, or mostly finished vitamin K product. The liver stores a bunch of the vitamins. So if you really want to get a bunch of vitamins, just eat some foie gras, eat some liver. Although foie gras is horrifying how they make it, but pate is pretty tasty. So liver's got a lot of vitamins. We also have non-processed versus processed grains, and there's of course degrees of processing, but like something like brown rice is going to be less processed, it has more fiber, vitamins, and minerals. But if you remove the bran in the husk, you're going to have less fiber, less vitamins, less man. That's why people say brown rice is healthier than white rice. Why would we even do this? Well, things cook faster, they need less water, they're easier to store, and some people like the taste because it's less chewy. So it's just like cheaper for companies to manufacture white rice than brown rice. So to offset this, a lot of things, cereals and whatnot are artificially imbued with extra vitamins. So we would call these fortified foods. Taking it a step further, in developing countries where people are just eating rice all day, they can't have a varied diet, right? So when I said most people are eating healthy foods or getting the vitamins, that's in the developed world. Like in a developing nation, somebody's very poor, eating only rice. They're not getting what they need. They're not getting enough thiamine. They're not getting enough B9 or whatever. Not enough vitamin A, and so. And it might be expensive and difficult, the logistics of how we're going to fortify all of their food. So we genetically engineer the food. So like the rice can produce beta carotene, which is a precursor to vitamin A. That can help cure vitamin A deficiencies. Now, this is kind of controversial, but also not something I want to get into. So good segue, beta carotene, vitamin A. So vitamin A, nicknames, retinol, retinoic acid. These all have slightly different forms and functions, which you can kind of ignore. Beta carotene is a precursor. I think the carrot is because we find a lot of it in carrots. Carrots have a lot of vitamin A. I was a kid once, and I was in training to become a soccer referee, and the guy who our teacher was, this ref, he brought in for lunch pizza and carrots, and of course all of the kids went to go grab the pizza. And then he scolded us because he was like, "Are you guys kidding me? Nobody grabbed a carrot. As a referee, your vision is everything, and you need carrots for good visions." And it was very silly, but it always stuck with me. So how does it help with vision? It teams up with opsin to make rhodopsin, which we can ignore the mechanism there. They just need that to transduce photons into chemical signals, which our brain interprets as images. It helps cells, specifically epithelial cells, differentiate. Somehow, above my pay grade, this is good for the skin. My dad's a dermatologist. Maybe I'll ask him. It's also a treatment for acute promyelocytic leukemia. So you don't really need to know the details too much about this, but this type of leukemia is like immature white blood cells. And they're just, of course, it's cancer, so it's massive in number. And rather than kill the cancer, what the treatment vitamin A does here, the all-trans retinoic acid, is it converts them into the adult form. And when they're in the adult form, they act very differently. They might kill themselves, and there'll be less division. So that's how this works, and it's just a good way to remember this. You also find this in a lot of acne creams. Vitamin B1 is thiamine. We already saw this. Tender loving care for Nancy. Tender, thiamine, TPP, pyruvate to acetyl-CoA is necessary. We see this actually twice in the Krebs in the making of acetyl-CoA. So with PDH, but then we also saw it in alpha-ketoglutarate dehydrogenase, which made succinyl-CoA. Anytime you're making a CoA, you've got a multimeric enzyme that needs thiamine. You also have it a little bit in the pentose phosphate pathway with transketolase, and we haven't talked about it, but branched amino acid breakdown also requires it as well. Alcoholics are particularly malnourished. The chronic alcohol abuse just inflames your GI, makes you really bad at absorbing vitamins, so less vitamin that way. And also, they're not known for having the most stellar diet, so they're just drinking a lot and maybe eating like candy or potato chips, or I don't know. They're probably not eating, like a nice hearty whole grain breakfast with vegetables and fruits and whatnot. So they are very likely to be vitamin deficient, especially in addition to people in developing countries. So if you have a thiamine deficiency, your mammillary body is damaged. It's caused Wernicke-Korsakoff syndrome. So Wernicke, I always thought of like Wernicke's area. It's kind of like, in the frontal lobe, and that's near the motor cortex. And I think of that as helping, being related to the ataxia. And, the frontal cortex, prefrontal cortex has to do with, intellect and advanced thought. And so this is going to lead to confusion. And there's also something called the frontal eye fields, which helps control eye movements. And so this is going to cause a ophthalmoplegia. So like an ophthalmologist is an eye doctor, and like a paraplegic, plegia means like a paralysis. So it's a paralysis of the eye. So this is a nice triad you'll see in Wernicke's. Korsakoff. So you have Wernicke-Korsakoff. Wernicke is one set of syndromes, and the Korsakoff the damage to the hippocampus in the mammillary body, which is associated with the hippocampus, it's going to cause retrograde and anterograde amnesia, hard time, remembering learning new things, and hard time maybe remembering old things. And what your brain does if you can't really remember something, your brain's constantly striving for coherence. So if I get up to go into the kitchen because I want a Coke, and I get to the kitchen, and then but I forget why I went there, and someone goes, "Hey, what are you doing in the kitchen? Why'd you come over from the living room?" And I don't know, maybe you're subconsciously embarrassed or something like that. Originally you wanted to Coke, but you don't remember that, and you might just say, "Oh, I wanted to see you," or "I wanted milk," or something, right? And it's your brain just striving for, grasping for straws to make sense of everything. So that is the confabulation aspect, which is secondary to the amnesia aspect. So if someone comes into you'll see a lot of, people abusing alcohol coming into the ER, and often they're hypoglycemic too, of course, because they're bad diet, they're just drinking a lot. You wouldn't want to give dextrose IV, which is just glucose. Why not? Well, because there might be a hypoglycemic, but there also might be thiamine deficient, and we need to do tests and figure out. But also better just to play it safe, because if you give somebody a bunch of glucose, what could be a problem if they're thiamine deficient? Well, this uses thiamine, so giving them glucose is gonna help, I guess, the hypoglycemia, but it's going to hurt the thiamine. It's going to exacerbate the thiamine deficiency, so that's problematic. So what we do is we give people like a banana bag IV situation, which has lots of electrolytes and sugars, but it's also going to have thiamine in here. Somewhere in here it says thiamine, or it should. Take my word for it, thiamine is something we give them. It's a banana bag. Fun side note, not really related, but ethanol is made into acetaldehyde with alcohol dehydrogenase, and then this is actually the very toxic thing. This is bad. This is bad news. This is eventually and slowly converted into, if you're drunk, you're slowly going to get less drunk over time, and that's because the acetaldehyde is converted into acetate. So this is, though, what makes you feel really shitty, and causes all of the symptoms of being drunk. So what an alcoholic could do is they could take something called disulfiram in the morning. So this is like if you have like willpower in the morning, but not willpower at night, right? So you're kind of like, you're doing a favor for your future self who doesn't have self-control. Because what happens is you inhibit this. So you take this in the morning, it inhibits that enzyme, which is mostly fine because you have breakfast, you have lunch, you go about your day. You don't really need this enzyme. But if you go to a party at night and you have this dilemma of, should I drink a bunch of alcohol, where you're going to feel like absolute shit if you do, because you're drinking the alcohol. This step works fine, but you're going to have huge levels of this that now, because you've destroyed this, or inhibited this enzyme, can't really do anything about. So it just makes drinking very unpleasant, because you get all of this nauseous, side effects and whatnot. So Wernicke-Korsakoff is one thing, but also beriberi, and I just think of B1 because we're in thiamine. People debate the etymology of this term. I cannot, or weak. Basically, people can become very, very weak. There's two different forms of beriberi. There's wet, which just means, or two different syndromes, I would say. Heart failure is going to lead to edema. And then the dry beriberi, we have muscle wasting and neuropathy. Vitamin B2. So you can think of FADH2 as being the link, or you can think of FADH2 as 1.5 ATP, which rounds up to 2. And that's kind of nice because it helps make a connection between vitamin B2 and vitamin B3. This is riboflavin. So remember that FAD is flavin adenosine dinucleotide. So the adenosine is one nucleotide, and the flavin is the second nucleotide. It's made in the Krebs cycle, the complex 2, the succinate fumarate, succinate dehydrogenase. And if you're deficient, you get angular cheilosis, which is just like inflammation and maybe get some scales at the corner of your mouth. Vitamin B3, so 2.5 ATP rounds up to 3, and this is again if we think about the name. NAD, nicotinamide, that's this guy right here. Niacin is specifically the nitrogen base. And then adenine is going to be the second. So this is the dinucleotide. This is made from tryptophan. Niacin is. So it's a tryptophan derivative. We can kind of see this aromatic structure here and then the aromatic structure in tryptophan. So it's not a huge leap. And randomly, it's really good for the HDL to LDL cholesterol ratio. Niacin's actually in tons of different stuff, so it's really difficult in a developing world if you're eating a normal diet to become deficient, as it is with many vitamins. But one way you could become deficient, one, I guess if you're an alcoholic, and two, if you're in a developing country, but if you have something called Hartnup disease, this is a renal malabsorption disease where I think it's like neutral amino acids have a hard time being reabsorbed from the filtrate back into the blood system. So you're going to shed and lose a lot of the neutral amino acids, one of them being tryptophan. And without tryptophan, you can't make niacin, and so then you're going to be B3 deficient. This causes pellagra, which we can assume, angular chelosis of B2. It's like, okay, you got some scaly corners of your mouth, but this is much worse, and it would make sense because, which one of these played a larger role from everything we've talked about metabolism? The 2.5 ATP, and we had a ton of them, or the 1.5 ATP, which we only made one that I can think of from the succinate dehydrogenase. That's, of course, an exaggeration. FAD is used for other things as well, but that's how I simplified in my dumb lizard brain. So pellagra, what are the symptoms? The 3Ds, dermatitis. Specifically, you get this, you can kind of see this little rash in the C4, C5 dermatome right there. They call it the collar rash. Dementia, diarrhea. Some people say it's the 4Ds because the fourth D is death. You can die from this. Vitamin B4, it's kind of like how Pluto used to be a planet, and then a bunch of people said, it doesn't really qualify as a planet. Except that I think they then went and said it is a planet again. I haven't been following that too closely, but adenine is obviously super important. We see it in ATP, NADH, FADH, right? It's the A that we're seeing here. And it's a DNA building block. It's so crazy important, but I think we can make it on our own, so it doesn't count as a vitamin. So vitamin B4 doesn't exist for all intents and purposes. Vitamin B5, 5-pento, as in 5-pentothenic acid, major enzyme of coenzyme A. So coenzyme A is right here, attached to this acetyl group, which came from the pyruvate, and the pantothenic acid is right there. Not that important to know about. Maybe know that you have dermatological issues if it's absent. Vitamin B6, I think of pyranose, which is a 6-membered ring, and this is B6, pure, and facilitates transamination and decarboxylation reactions. Transamination, where do we see amino groups? Well, in the amino acids. So it's going to make sense that it's going to help converting from one amino acid to another, and that's also going to be playing a role in, amino acids can be converted into a lot of interesting downstream products, we can make melatonin, serotonin, histamine, heme, all of these cool neurotransmitters are dependent on it. Also, it's a decarboxylase, or it's associated with them. So breaking down of glycogen is going to be important as well. Isoniazid is a tuberculosis drug, and oral contraceptive pills. These can lead to deficiency. And if you have a deficiency, you might have neuropathy, so tingling in your extremities, and confusion. And I just think of that as being, you need B6 to get a lot of these downstream, catecholamines and whatnot. So, sure, you're going to have some weird neuro symptoms. That seems, I buy it. And then hemosiderin anemia is a type of anemia where you're effectively not making heme. And again, that's because we need B6 to make this heme. So we're going to have an anemia there. B7, biotin. This is associated with any sort of carboxylation reaction. So carboxylation reaction means we're adding a carbon to something. When would you want to add a carbon to something to? Well, if you're making a fat, fats have tons of carbon. So if I'm making a fat, I'm building carbons on carbons on carbons, I need B7. Additionally, gluconeogenesis is you're making a glucose from, a new glucose from scratch, right? From a bunch of random stuff. So you're going to be building carbons and carbons a lot. So it kind of makes sense that it would be involved in these two pathways. Again, if it's deficient, dermatological issues, I would know that it reacts very strongly with streptavidin and avidin. I forget what, maybe streptavidin has higher affinity, but they're effectively the same thing. Here, pyruvate becomes oxaloacetate via the pyruvate carboxylase, and then eventually that's put in here for gluconeogenesis. We'll talk more about that later. We see that here. Biotin is also used in a lot of labs to tag a protein. So you can biotinylate a protein, which just means, here's my protein here, and then here's my protein, but now we've attached it to a biotin thing. Why is this useful? Because remember that avidin, streptavidin has a really, really, KD is 10 to the negative 15, crazy high affinity. They can grab onto this biotin and we can immunoprecipitate, we can pull it down. On that note, there's a lot of avidin in egg whites. And if you cook them, it's fine because it'll just denature and scramble and it can't really do its job. But if you're just drinking a bunch of raw eggs, especially if you're drinking a bunch of raw egg whites, you're going to have a bunch of avidin going into your body. And that avidin is going to circulate and it's going to chelate. It's going to strip all of the biotin from your body. It can make you really sick. B8 inositol, we see this in a receptor tyrosine kinase pathway. It's a phosphatidylinositol is a whole signaling thing and blah, blah, blah. We'll get into it, but it's obviously very important, but I suppose it doesn't count as a vitamin because we can manufacture it. B9, folate. So we'll get into this later, but this has a lot to do with pregnancy, and I think of 9 as the 9 months of pregnancy, and folate has to do with pregnancy. Also called folic acid. The active form is tetrahydrofolate, THF. It can also be methylated. That's kind of the whole point. And sometimes they'll say annoying things like 510 or tetrahydrofolate with a methyl group, or just 5 tetrahydrofolate with a methyl group. You might have to know that. So it's involved in the methylation pathway, which we're going to talk about here. And I said that this is an anachronistic lecture, and that's because we're also going to talk about methylation in a future lecture as well. But we'll touch on it here. So when do we need to methylate things? Uracil to thymine, the RNA nucleotide to its identical DNA nucleotide, except that, I guess we've taken off one of the OHs on the ribose sugar, but just looking at the nitrogen base, here's my uracil and here's my thymine, and boom, we put a methyl group there. This is going to be really important for cells that are dividing rapidly, which is mostly the hematopoietic stem cells, which make red blood cells and white blood cells. Also, ethanolamine to choline. You don't really have to know this too much, but you just see here, there's a bunch of hydrogens here, there's a bunch of methyl groups. Why is this important? I would just know that myelin has this molecule which must be methylated, and so we'll see what happens if that goes awry. Remember, foliage is in a bunch of green leafy vegetables. B9 and B12 are going to kind of be in cahoots together. They're working. This is a very complicated picture. You can probably ignore this for the most part, but this is maybe, if you're trying to better understand this might be a good thing to come back to really dig in and get a great understanding. But, we have the folate cycles coupled with methionine cycles, coupled with the sulfuration cycle. Simplification, it's really complex. It's really important to know. We're only going to go into it a little bit here. We'll talk more about it later. But folate, B9, becomes THF, the active form. THF can be methylated. Now that methylated THF has 2 different options. Option number 1, it can go this way, and it can help convert a uracil into a thymine, which is really just a uracil, with a methyl group attached onto it. Well, that now gets rid of the methyl, and now it's just THF again, and it needs to be regenerated. Now, first of all, how do we get the methyl on? Serine to become glycine. There's an extra, it's the R group here is a carbon group connected to an OH. Ignore the OH, but just the idea is we're losing one of our carbons going from serine to glycine. And anytime we're doing a conversion between 2 amino acids, we do need B6. That's not super high yield, so if you're going to ignore anything on this slide, this is a very high yield, but I would ignore that if you limit it on brain space. That's how we regenerate. That's how we put the methyl on. I would know that. But anyways, back to the main plot. This is one thing that THF CH3 can do. Another thing it can do is pair up with B12 and methionine synthase. Most importantly, let's focus on B12. And blah, 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 blah. We'll do a bunch of this other stuff later. Converts homocysteine to methionine. The important thing is that we're going to take a methyl. We're going to put the methyl elsewhere. We're going to give it to the SAM who carries the methyl. So we're passing the torch onto the SAM. And that can, theoretically, actually, this could also convert uracil to thymine. But I think it does a lot of other methylation things that are above my pay grade. One, though, that it does is ethanolamine to choline. So again, THF-CH3 can just give it directly to the nucleotide, or it can help give it to the B12, which gives it to the, makes the methionine, which gives it to the SAM, which can methylate other things. Okay, so there is a little intermediate here, which is important because if you are missing B12, sometimes we go route number 1 and then sometimes we go route number 2. Well, if you decide to go route number, let's call it 50-50. If you decide to go route number 2 and you don't have any B12, you are stuck as this intermediate, which can build up and is problematic. What can happen if you're deficient? So let's focus mostly on the fact that we cannot make thymine, so we cannot do a lot of DNA synthesis in cells that are rapidly dividing. That's mostly hematopoietic cells, and yeah, so when they're not dividing, dividing in the hematopoietic stem cells actually makes them a little bit smaller. So you get these large red blood cells, these macrocytes, and we don't really have a point of reference, but these red blood cells are, all rather large. You also get these hypersegmented neutrophils. So there should just really be 3 lobes to for the nuclear segments, but here you have like 6 or more. Sometimes these are called PMNs, polymorphonuclear segment. I just screwed that up, but it's PMN is just another fancy name. We see this right here. Folate B9. Remember 9 for the 9 months of pregnancy. There's a huge demand for B9 because there's lots of cell division to make a baby. So we need a lot of B9. And B12 is, you don't need as much B12, I suppose, for whatever reason. Maybe it's because we saw here that you theoretically could do this without B12. That's a guess. I'm not really sure. But folate is the most significant one associated with pregnancy. So a lot of people will just go ahead and take, they have pills just for pregnant ladies to help their baby. So what happens if you don't, you can get lots of neural tube defects. So I guess there's a extra need for cell division for the nervous system and the spinal cord. And so you can get spinal cord and the meninges, which kind of stick out, and it's called spina bifida. You can get other things too, but we just know spina bifida is associated with folate deficient women who are pregnant. PABA makes folate. Bacteria do this. I don't know. Somebody figured out that this doesn't count as a vitamin. Salicylic acid. We see this in acnes and creams. Precursor for aspirin. It's also a plant hormone. By the way, this is just for fun. You don't have to know any of the ones that don't count as vitamins. I just thought that was interesting for me. Cobalamin. I think of Kobe beef, cobalamin. This is in lots of meats. Let's revisit everyone's favorite methylation cycle. This folate becomes DHF, which we didn't talk about as an intermediate, which becomes THF. Serine to glycine puts on a methyl. Sometimes we call this 5,10-methylene THF. This can go to the DNA synthesis pathway, converting U to T, which we need for cell division, or it can make this little intermediate here, which just passes the torch on, with the assistance of B12. Homocysteine becomes methionine, so there's an extra methyl there. That gives the methyl to SAM, and then SAM can methylate DNA as well, but often it's going to just, I think, methylate various proteins, and something like ethanoline to choline, which is, again, for choline as well, I think of choline as, we see CH, choline. So I think of all of the methyl groups on there. And again, that's important for myelin. As a random, but it's also involved in the conversion of, so this is not at all related to the previous pathway. Methylmalonic acid is some random molecule. It needs to be broken down into methylmalonyl-CoA, which is eventually broken down to succinyl-CoA with the help of B12. Now, who cares? Why is this significant? Because how do we distinguish B9 from a B12 deficiency? They're both very similar. You need both of them to have this methylation pathway run. The B9 and B12 deficiency, if I don't have this and I don't have this, I cannot convert this into this, period. So the homocysteine levels are going to go up. They're going to go up and up and up and up. That's in both of them, right? Homocysteine and homocysteine levels are up. But only the B12 is involved in this pathway. And if I don't have a B12, then I'm just going to have a higher level of methylmalonic acid. So the B12 is homocysteine and methylmalonic. So you can think of that as like 1 and 2, right? So it's like 2 different things, the homocysteine and methylmalonic acid. Or you can just think of 1, 2, 12. The 2 stands for that it's both pathways. I don't care. Figure it out. It's very important. That's crazy high yield to know. So when cobalamin, methylation pathway, uracilthymine, hematopoietic stem cells, ethanolamine to choline, that's for the myelin, and cobalamin, Kobe beef, this is the meat, dairy, eggs. So in other words, vegans have an issue with cobalamin. Now, folate has only stores for a couple of weeks. You run out of folate, which is B9, pretty quickly. In addition to the increased demand, that's why pregnant women might have an issue. But cobalamin is there for years. So even if someone's a vegan, let's say you're fat, or you're like, this week I'm doing intermittent fasting, and then next week I'm going to be a vegan, and then the next week, and you might say, oh my gosh, I did 6 days of not eating meat. I'm B12 deficient. You have years of stores of B12. So you have to be really strict, because even if you eat just meat or dairy or eggs every once in a while, you're going to replenish those B12 stores. So it really have to be a very strict, lifelong vegan. Um, so yeah, very strict, long haul vegans. Pernicious anemia. So B12 absorption. This picture is kind of confusing, but go ahead and pause it and take a look at it and make sense of it. B12 to be absorbed in the small intestine, specifically in the ileum, requires this molecule called intrinsic factor, which is made by the parietal cells in the stomach. So the stomach makes the intrinsic factor. Intrinsic factor binds to B12 and facilitates later in the small intestine its absorption into the body. Pernicious anemia is where people have this like autoimmune syndrome where they destroy all of their parietal cells. So they cannot make intrinsic factor, which means they cannot absorb B12. They're going to be deficient. Similarly, if someone has Crohn's disease, or ulcerative colitis, or that they can screw up their ileum, or if you have like GI surgery, maybe you're getting gastric bypass, well, that's also going to affect your ability to absorb B12. So it causes the same symptoms here. So it can cause the macrocytes, the large red blood cells, and then the hypersegmented PMNs for the neutrophils. I mentioned it can cause the spina bifida, but because we have lots of stores, and for whatever reason it doesn't seem to be as linked, it can cause neural tube defects, but it's less likely. So what's unique to B12 that we don't see in folate is that we have demyelination. And again, that's because remember that we're making choline, which is in myelin, and so that's the link. It can be in the periphery, so like the fingers and toes, but also in the spine. And when it's in the spine, there's a fancy pants term called subacute combined degeneration, which just means your spinal cord is going to be demyelinated, which is problematic. And it's actually more than that as well. So it's not just the demyelination. They found that folate supplementation can actually exacerbate the issue. So they think what happens if you don't have B12, we mentioned earlier, you can get a buildup of this. And this might be toxic for, I think it's like cells in the dorsal root ganglion, which you don't really have to know. But if you keep putting in folate, you might increase the levels of this, which exacerbates the toxicity. Additionally, it's the case that if we don't have our B12 here, we're going to have a buildup of methylmalonic acid right there. And that also might somehow lead to toxicity that is unique to B12 that we don't really see in B9. So again, the buildup of those, in addition to the fact that we can't make choline, might cause this demyelination and nervous system issues. Whew, okay, we did it. We're out of the B's, home stretch. So vitamin C, ascorbic acid, is it's involved in the glutathione pathway. So it can oxidize glutathione. So it's really just carrying the torch, right? Glutathione is reduced, and it's going to oxidize, a nasty reactive oxygen species, a nasty peroxide. But for whatever reason, vitamin C says, hey, I'll take the extra electrons and I'll reduce the hydrogen peroxide. I don't know why that is happening or why glutathione can't do it alone, but that's a thing. And remember that it also, from the last lecture, plays a role in collagen production. So we have hydroxylases, which, collagen is this glycine, proline, X repeat. Often that's the case. And then the proline itself, it could be a lysine, but it's often a proline, can be hydroxylated so that it can, we give an OH to it so that it can then grab onto a sugar, and somehow that helps with cross-linking. In any case, so once you hydroxylate, a lot of enzymes just get tired out, right? The whole idea of an enzyme and catalyst is that it's regenerated. Most of them are automatically regenerated. Some of them, though, need a little bit of help to be regenerated. And here, ascorbic acid is going to work through iron to regenerate our enzyme here. So long story short, you need it for healthy collagen. It's in lots of fruits and veggies. We've seen it, Sunny D, orange juice, how many things advertise how much vitamin C they have. This is like an absurd amount of vitamin C, more than you'd ever need. Also an important point, somehow, I'm not really clear on the link, somehow vitamin C is linked to a healthy immune system. So maybe know that. And this is just a fun picture showing the NADPH interacting with the glutathione. So this is kind of like when you have cysteine disulfide bonds. This is two glutathiones and the oxidized version together, and then we can reduce them. But then we can just pass the torch on to vitamin C, and then it's not really passing the torch because it's kind of the end of the line. This is the point. We are dodging a bullet. We are saving the cell by converting a nasty hydrogen peroxide to water so that it doesn't damage like a nucleotide or a protein or something. Vitamin D, calcitriol, because it helps with calcium. You remember ADEK, ADEK. These are our nonpolar vitamins that you can go right through the membrane. It acts kind of like a testosterone or progesterone or aldosterone hormone. It goes through, it binds to an intracellular receptor, and then it can act as a transcription factor. Now it's a little bit more complicated than that. And so it can come from meat, dairy, eggs, and the sun. Let's look at the sun. The sun can turn cholesterol into vitamin D, but that needs to be hydroxylated, so it's not quite active yet. 25 hydroxylase in the liver, but then it's going to go to the target tissues, which are going to be the bone, the gut, or the kidney. So here they're showing the kidney. It can go to the kidney. When it goes into the cells, the 1-alpha hydroxylase finishes the job in the cytoplasm. Now it's mature. Now it can bind to the transcription. So we see this here, 25. And it goes in, so here it's in the blood, goes into the cell, the 1-alpha hydroxylase makes it the mature, it's now going to bind onto a receptor, it goes and it helps regulate transcription. What does this do in cells? Well, just remember that vitamin D is trying to increase the calcium in your blood. So what are a couple of ways to do that? Well, your bones have tons of calcium. Osteoclasts can do bone resorption, which means they break down the hydroxyapatite matrix of the bone and they push calcium into the blood. So we can stimulate osteoclasts, which is going to break down the bone, push calcium into the blood. Just help us reabsorb or absorb more calcium from the GI, because you might not be getting every bit of calcium normally, but if I have a lot of vitamin D, maybe it's make it really efficient and effective the amount of calcium I'm getting from XYZ food source. And then also you pee out a fair amount of calcium, but you know what? If we go to the kidneys, we can go to the tubular cells and say, hey, like, can you not piss out so much calcium? We'd like to reabsorb more of that, and they'll do that. They also, I would note, it's kind of the opposite. They're going to piss out more phosphate, and so you reabsorb more calcium and you lose more phosphate is often something that vitamin D does in the kidneys, and that might be where its most significant effect is in the kidneys. Quick note on this real quick. So we said that one way to get more calcium is by osteoclasts breaking down bone. That means that osteoclasts, which are really just macrophages. So, I thought of like chicken of the sea. We kind of have like this is like a macrophage of the bone. Macrophages of the bone are osteoclasts. Macrophages of the brain are microglia. Macrophages of the liver are Kupffer cells. So we have a lot of different names for macrophages, and this is one you should know. So they have 1-alpha hydroxylase to activate the vitamin D. It makes sense when we talk about the purpose. Sarcoidosis is a disease where people basically have these large granulomas, and there's lots of macrophages and giant cells in there, and they're going to have lots of 1-alpha hydroxylase. So someone comes in with sarcoidosis, what might be one of the ways we can tell is they're going to have a lot of 1-alpha hydroxylase, which means they have a lot of vitamin D, which means they're going to be hypercalcemic. Any case, so what happens if I have a vitamin D deficiency? I don't have enough calcium. If I don't have enough calcium, I'm not going to have proper bone development. So crickets in kids, and you can see these bow legs are going to be a common clinical presentation. And then osteomalacia, which is just kind of like bad, shitty bones in adults. Vitamin E, tocopherol and tocotrienol. This is just like vitamin C. It's just like one extra thing, right? So the NADPH can the glutathione, passes the torch to the vitamin C, passes the torch to the vitamin E. Vitamin E can go ahead and reduce a peroxide to a water, but for whatever reason, and it's actually important, it often works on lipids and membranes. So this is going to be especially important in red blood cells and also in myelin, which is, of course, just tons of membrane, proteins and lipids coupled together. So if you're deficient, this explains the symptoms. The red blood cells, which are particularly prone to oxidation because of all of that oxygen, can have hemolytic anemia. So the red blood cells will explode and die, and that's because there's oxidation of the membrane of red blood cells. And then peripheral neuropathy. So you're going to get tingling in the fingers and toes because you are destroying, or the oxidation of the myelin lipids is not being reversed. Vitamin K, so K for coagulation. It's actually, I think, the initial German spelling. So this is associated with blood clotting. So specifically, it's going to carboxylate the gamma carbon of a glutamate. So, if I have a C double bond O, here's my amino acid N R. All right, so here's our amino acid, and this is an alpha carbon. This is our beta carbon, and then this is our gamma carbon over here, and our gamma carbon now has a COO minus. This is normally what a glutamate looks like right here, and then the vitamin K is helping us carboxylate, putting another one of these things here. Much prettier picture is shown right here. Here's a glutamate on a protein. We have our alpha not shown, beta, gamma, and then we're going to put another glutamate on it here so that it can do cool, fun stuff with calcium, which we don't have to worry about. It helps us do blood clotting so that if I get cut, I don't die. You also get it from green leafy vegetables like B9, like folate, but remember that the bacteria in your GI system are going to help make the finished product. And if you're deficient, you're going to have lots of bleeding and bruising and bleeding in your stool, and so it's going to look black and tarry. Now, a note about this. Remember, we said that there's a lot of enzymes that just kind of get tired after they've done their job once, and we need to rejuvenate them. So vitamin K is one of those. So vitamin K, maybe it's not so much the enzyme, but vitamin K itself gets used up here. Like vitamin C, like vitamin E, it gets used up. We need to regenerate it. So we have epoxide reductase, which is going to regenerate it. I would definitely know high yield that warfarin is a drug which is associated with clotting, can go ahead and inhibit epoxide reductase, which means that you can't get your vitamin K back and you're going to have clotting issues. Which is actually its intended effect. All right, let's do it. Let's jump in. Vitamin A, retinoic acid, beta carotene. Think of carrots. It helps with vision. Remember that silly soccer referee. That's because it combines with opsin to form rhodopsin, which interacts with photons. It also helps differentiation, right? And that's, remember, like retin-A micro, and tretinoin. A lot of these acne medications have vitamin A because it helps with skin cell differentiation. I don't know how that works. It's also used for APML to help differentiate the cancer cells so that they're not a cancer anymore. I don't actually know how the differentiation occurs. For food, ignore all of this stuff. We're just going to focus on this one, and this one, and this one. Ignore everything else. Ignore. All right, so ignore the food. I can't think of anything that depletes it. Causes vision issues if deficient. All right, let's look at B1. B1 is thiamine. This is remember, tender, loving care for Nancy. Tender. We see it in PDH. We also see it in alpha-ketoglutarate dehydrogenase. It's also a transketolase in the HMP shunt, which we haven't seen yet. We also haven't seen the branched amino acid pathway, but it's there too. Alcoholics, not known for their best diets. Also inflammation of the GI, affects absorption, causes Wernicke-Korsakoff. Remember that Wernicke, I think of like Wernicke's area. This is the frontal lobe one. You have ataxia, ophthalmoplegia, and you're also going to have confusion. And then berry, we have wet and dry. Wet is going to be heart failure, edema, and then dry is going to be your neuropathy and your muscle wasting. Korsakoff, I forgot to mention, is off your rocker. This is where you forget things, old. You can't learn new information, you can't remember a lot of old stuff. But then you confabulate to, try to, your brain's trying to make the world coherent and make sense. All right, B2, think of FADH2 or the 1.5 ATP, rounds up to 2, riboflavin. It's a dinucleotide, it's an electron carrier. We see it in succinate dehydrogenase, which is complex 2 at the Krebs cycle and electron transport chain. Not much to know about it. You get angular chelosis, the angles of the mouth, you get kind of inflamed and chapped and whatnot. B3, think of 2.5 ATP for NADH, rounds up to 3. Nicotinic acid, and it's a tryptophan derivative. Electron carrier, all over the place in glycolysis and Krebs and whatnot. Remember that it has this weird effect where it lowers LDL and it raises HDL. Heart-Nup disease, so it's hard to become deficient, but Heart-Nup disease is where you can't reabsorb tryptophan in your kidneys, and tryptophan is the upstream product or the upstream precursor, and so you don't get any niacin. It leads to pellagra, the 3 Ds, dermatitis, diarrhea, dementia. The 4th D sometimes is death. B5, pantothenic acid. It's coenzyme A. It involves this structure. It's in it's in the middle there. It helps with acyl transferases, like we see with PDH, like we see with alpha-ketoglutarate dehydrogenase. and you get dermatology issues if it's deficient. B6, think of pyranose. Pyranose is a 6-membered ring. That's his B6. Transamination, so converting amino acids to oxo acids, which is really just interconverting one amino acid to another. We'll talk about this in a future lecture. Also, changing amino acids, similar note, making downstream derivatives like a lot of the catecholamines, norepinephrine, epinephrine, making heme from glycine. It also plays a role in the phosphorylase for glycogen breakdown. And remember that, if we're breaking down glycogen, we must be decarboxylating. Isoniazid and tuberculosis and oral contraceptive pills can deplete it. And if you don't have heme, you might have an anemia. And if you don't have neurotransmitters, this might cause some sort of neuropathy. B7 is biotin. It causes carboxylation. When am I building carbons up? Well, if I'm making fat or if I'm making new sugar. If you have raw eggs, maybe we should know this one as well. In the egg whites, there is avidin. Avidin has a crazy high affinity for biotin. So if you're just chugging raw eggs trying to get really fit, you're going to make yourself sick because it's going to chelate all of the biotin. Also causes derm issues, not much to write home about. B9, very important. B9, I think of the 9 months of pregnancy. Folic acid, also known as folate. THF is the active form. It can be methylated. So sometimes it's THF-CH3. Sometimes they say N5, N10, or just N5, and maybe you should know that. We can methylate homocysteine to methionine with the help of B12, or not using B12. We can just go straight up and go uracil to thymine. Which is really important for DNA synthesis, especially in hematopoietic stem cells, which are dividing all of the time. The methylation cycle, though, also can make choline. Think of ethanolamine to choline. Choline has a lot of CH, and that's going to be a component in myelin, and so this is going to affect nervous system structures, which we'll get to. Folate, foliage, green leafy vegetables. A lot of people take prenatal vitamins when they're pregnant because you're making a baby, so that requires a lot of cell division, apparently, right? One cell becomes 40 trillion cells. So we definitely need a lot of folate. That's why we take the things. Anti-seizure medicines like phenytoin and then methotrexate, it can be used for autoimmune diseases. It can be used for, this is a chemotherapy agent. It's going to be like a folate like mimic, and it's going to antagonize it, and it can lead to lower folate levels. And there's limited stores of B9, which is, again, one of the reasons that you might become deficient. So pancytopenia, we can't make red blood cells. We can't make white blood cells. We have trouble making platelets. You're going to have anemia. You might get sick a lot. You might get tired really easily. The anemia is megaloblastic. We have these really large red blood cells. And and we have these PMNs, which is just like a hypersegmented neutrophil. And you get neural tube defects if you're pregnant and you're B9 deficient, potentially. B12, you can think of 1 or 2, or you can just think of the fact that there's a 2 there, and that is to remind us that this is 2 different pathways, right? There's the methylation cycle, but then also randomly, we have the methylmalonic acid pathway breakdown. So if we have B9, we will only have a homocysteine increase. If you have a B12, you'll have a homocysteine increase, and you'll have a methylmalonic acid increase because you can't break it down. B12 is really just assisting here. It's not carrying it as much as the folate. Kobe beef, it's in beef, it's in meat, it's in dairy, it's in eggs. So people who are vegan would have a hard time with this, only if they're a lifestyle vegan, though, because we have lots of stores for years. Pernicious anemia, some people have an autoimmune attack of their parietal cells, which makes intrinsic factor, which is necessary to bind to B12 to be reabsorbed effectively in the ileum of the intestines. Also, some people have like Crohn's disease or other autoimmune GI things, and it's hard to absorb nutrients because you're damaging the lining of your intestines. If you get gastric bypass or other surgery, that might also make it harder. We similarly get a pancytopenia, which means we'll have a megaloblastic anemia. The PMNs and whatnot. Neural tube defects, not as much, but it can still happen. Subacute combined degeneration, fancy term for just demyelinating your spinal cord, but it actually might be more than that. It might be the dorsal root ganglion are dying because we're trapping the methylated B9 intermediate, just the 5, the 5 N5 THF-CH3. It also could be the increase in methylmalonic acid. Also, the peripheral neuropathy is kind of associated with that as well, and so you're going to get tingling in the extremities. Ascorbic acid, vitamin C, drink a lot of orange juice. This is going to, the baton has passed from glutathione to vitamin C, and so vitamin C can then, deal with those pesky peroxides. Can also regenerate hydroxylases and that's necessary for collagen, specifically like proline, maybe a lysine hydroxylase. There's an antioxidant, which is why some people might want to put it on their skin, and collagen synthesis. Somehow it's linked to the immune system. People say drink a lot of orange juice when you're sick. So it's in fruits and veggies, hence orange juice. Pirates had an issue with this because they're just they're bringing jerky and biscuits on to a ship for months at a time. So they're not getting enough vitamin C. So scurvy, I think of a pirate. You get swollen gums, easy bruising, anemia, poor wound healing, corkscrew hair. There's a lot of collagen in hair. All of this has to do with defective collagen. Also vision issues. Because there's a lot of collagen in your eyes. Calcitriol, think of calcium with vitamin D. We're trying to increase the amount of calcium. It acts like a non-polar hormone, so it's gonna, remember it's got to be hydroxy. We can get it from the sun, so we can get it from dairy fish and the sun. Cholesterol is converted to vitamin D, but it's inactive. It needs to be 25 hydroxylated in the liver, and then it goes to the target tissues. What are the target tissues? Osteoclasts in the bone, tubular cells in the kidneys, or what's the last one? And then your small intestine, brush border cells. So it's going to have the 1 hydroxylase there. So it's got to be 125 hydroxylated to work effectively. And once it's activated, it's going to bind to a receptor. It's going to act as a transcription factor and change the regulation. How does it work specifically? We want more calcium. So we're going to breaking down bones with osteoclast. We're going to reabsorb more calcium from the filtrate from the urine back into the blood, and we're going to absorb more calcium from our diet in the GI system. Remember that osteoclasts are basically macrophages, and they have the 1 alpha hydroxylase to activate vitamin D. Sarcoidosis is a disease where you have lots of macrophages and granulomas, and so you will have excess vitamin D, and you will therefore have hypercalcemia. If you're deficient, though, you could get rickets. You have bow legs as a kid, and then you just have bad bones, osteomalacia as an adult. Vitamin E, tocopherol and tocotrienol, I'm sure I'm mispronouncing those. Really just kind of like vitamin C. It's the baton is being passed from glutathione to vitamin C and from vitamin C to vitamin E, and it's going to reduce a bunch of random stuff, but especially lipids in the red blood cells, but also lipids in the membrane of myelin. It's an antioxidant. And so what happens if I damage my red blood cells because I'm oxidizing the membrane and I don't have any vitamin E to reduce it, to bring it back to normal? Hemolytic anemia, and then you might get subacute combined degeneration and peripheral neuropathy because you're really just damaging the myelin. So you're going to get like neuropathy issues. Your myelin's getting screwed up, and no vitamin E to reduce it. Vitamin K, as far as I know, there's no nickname I'm sure there's tons of little names, but I don't know if you're responsible for or it's high yield to know the nicknames there. Gamma carboxylation is what vitamin K assists with. Remember that it has to be regenerated by epoxide reductase, but warfarin can inhibit that. Vitamin K for coagulation. It's going to help mature a lot of our clotting factors. It's in green leafy vegetables like folate, but it has to really be made at the end of the day by our GI bacteria. So warfarin can inhibit it, but also non-specific antibiotics. I didn't mention this earlier, but that's important. If you have non-specific antibiotics, which means it's broad spectrum, it's targeting lots of bacteria, that can damage the bacteria which are making the vitamin K, the good bacteria in your intestines, so you're going to become vitamin K deficient. So excess bleeding, easy bruising, maybe black tarry stools. Gluconeogenesis, if we really break down glucose, new, genesis, the making of something, is that we're making new glucose sort of from scratch and pushing it out into the bloodstream to feed the rest of the body. And you could have guessed it, who's behind this? The liver. That altruist is making sugar, so many metabolic things. Really, you just think metabolic pathway probably is happening in the liver. That's at least a good guess. And I think of a MacGyver here because we're really just taking random stuff, glycerol, amino acids, lactate, whatever we can get our hands on, and we are converting it into, somehow feeding it into this pathway to make glucose, right? So MacGyver was really good at taking here's a rubber band and a pen and I made a slingshot, and here's a bing bang boom and I made a whatever. So that's at least how I thought of this. It's really just glycolysis backwards. Glucose becomes pyruvate, or now we can go backwards, pyruvate becomes glucose. Now, there were these three irreversible enzymes, 1, 3, and 10, sometimes called 1, 3, 9, for glycolysis. So we're going to need new enzymes there. So instead of hexokinase, we're going to have glucose 6-phosphatase. Remember, you're typically named after the reactant. So here, glucose 6-phosphate. The reactant is this, glucose 6-phosphate. And it's a phosphatase. It's using a water to break off the phosphate. Here we had phosphofructose kinase, PFK1. Here's F16, the reactant, bis-phosphatase. We're taking off the phosphate. The weird one is pyruvate kinase, where we have two separate things, which we'll actually get into in a little bit. But we're really going to make oxaloacetate, OAA, with pyruvate carboxylase. And then we have a second step where we do PEP carboxykinase to make phosphoenol pyruvate. And remember, this is the liver, and we have a lot of different glucose transporters. This is specifically going to be glucose 2 in the liver. It's bidirectional. Why? Well, because sometimes you had 10 brownies, you have too much sugar in the blood, it needs to go into the liver just because it's toxic, right? That's the reason it comes into the liver. Hence the high Km of the GLUT here and of the glucokinase versus hexokinase. But again, the liver's job is to push glucose out a lot, right? If you're starving, you've been, stranded in the woods for a couple of days, haven't eaten anything but a couple of twigs, your liver is going to do glycogenolysis, which we'll talk about, but also gluconeogenesis. And to do that, it's going to make new glucose or break down glycogen into glucose, and it needs to push it outwards. So this outwards movement is unique, and that is why glucose 2 is here, and I just think of the 2 as a bidirectional feature. So there, this is a little bit more detailed picture. The reason it's a MacGyver, again, is we have a lot of different inputs, and they don't all start at pyruvate. They can come at random times. So we have glycerol, we have lactate coming in, that does go straight to pyruvate, and then amino acids can come at various steps and various points. And again, this is going to be a little bit more complicated. We'll get into with ketogenic versus glucogenic. So starting with glycerol. So again, glycerol is attached to 3 fatty acids for triglyceride. This is really just what an adipose tissue is, a fat cell. It's just a bunch of triglycerides. If you have epinephrine or glucagon, you're going to break down this, and the fatty acids go, and we can do beta oxidation with them, more on that in a future lecture. But then the glycerol component, this is the ugliest picture I've ever made, but the glycerol goes into the blood, hence this red stuff, and it moves and it moves and it moves, and it goes to these hungry, energy-depleted cells. It goes in there, and it can be converted into DHAP, which can be, fed into glycolysis in a normal hungry cell, or in the liver, it can be fed into the gluconeogenesis. So instead of going down glycolysis, it goes up through gluconeogenesis to make a new sugar, which then goes back out into the liver and feeds the rest of the body. The Cori cycle, so here we're looking at lactate. So we can take lactate, bing, bang, boom, MacGyver, we turn it into a pyruvate, turns it into glucose, that's gluconeogenesis, put that out in the blood, feed the hungry cells. Who needs a lot of glucose? Well, the muscles, especially if I'm exercising, and I might have more energy demands than the oxygen can meet up with. So I switch over to lactic acid fermentation because I want to regenerate that NAD plus. And so we're going to generate a lot more lactate. Additionally, red blood cells, they're going to take a lot of glucose, and they can only ever do anaerobic respiration because they don't have mitochondria. So in both these situations, we're making a lot of lactate, which we can then ship back again. So this just cycle of gluconeogenesis in the liver makes glucose, which can be anaerobically converted into lactate and sent back to the liver, is the Cori cycle. So as we mentioned, pyruvate, we have these 3 irreversible enzymes, and this last one here, it's kind of weird. For whatever reason, we don't just go from pyruvate to phosphoenol pyruvate. We first make a pit stop at oxaloacetate, OAA, and then that becomes the phosphoenol pyruvate. So we see this here. We have a 1, 2, 3 carbon, and randomly 1, 2, 3, 4 carbons. We carboxylate, hence the name pyruvate carboxylase, adding a carbon group. Whenever you add a carbon group, like with fatty acid synthesis, or we talked about earlier, gluconeogenesis, this is it. This is where biotin B7 comes into the picture. It's weird. You put on that carbon only to immediately, so plus a carbon, only to immediately minus a carbon to make it back. So phosphoenol pyruvate carboxykinase. And let's just take a second to appreciate how amazing pyruvate is. Gluconeogenesis to glucose in the liver cells. All cells can do glucose to pyruvate and glycolysis. Anaerobic respiration makes lactate. You can then put it into the the matrix and make acetyl-CoA for the Krebs cycle. You can make oxaloacetate and I guess technically it's the oxaloacetate to glucose and gluconeogenesis. And then we can make alanine with our transaminases, which are fueled by our B6 vitamins. So pyruvate can really, is a nice hub that can connect to a bunch of different metabolic intermediates. On this note, though, of pyruvate becoming oxaloacetate, this is a nice link between the ketogenic process. So we've seen that gluconeogenesis, we can put in glycerol, we can put in lactate, and we can put in amino acid. But there's a big asterisk there because it's really complicated. Different amino acids go into this process at different points. We can separate this into two different things. Those that can become acetyl-CoA, this is a ketogenic amino acid. So if you can be converted to acetyl-CoA, you're ketogenic. Anything else, alpha-ketoglutarate, succinyl-CoA, fumarate, oxaloacetate, pyruvate, all of those are glucogenic. And some of them, as we can see, can do both, right? Like tryptophan is right here and tryptophan is right there. The only two that can only do acetyl-CoA are the lysine and leucine, K and L. So let's understand why making an acetyl-CoA. So acetyl-CoA can make, ketogenesis can make a ketone. So that's not too crazy. You can make a ketone with acetyl-CoA. But the real question is, why so acetyl-CoA can become a ketone, but why can't acetyl-CoA become a glucose, right? So pyruvate can become glucose because pyruvate, like we just saw, can be carboxylated to oxaloacetate, that weird intermediate, which can become PEP and goes back to glucose. So that's one down, that makes sense. Let's look at these. So imagine you, for the Krebs cycle, we just have one oxaloacetate, just one OAA. That's all we have. We don't have any citrate, no isocitrate, no alpha, no blah, 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 blah. We only have this one. I'm not saying we have one of everything. We only have one OAA. So what happens if I take an arginine and convert it into a glutamate and make it into an alpha-ketoglutarate? Well, I now have one of this, and then I'm going to lose that because I'm going to gain one of this, and I'm going to lose that because it's going to become that, and I'm going to lose that, I'm going to get one of that, and I'm going to lose that, and I'm going to make one of this, and I'm going to lose that. Now I have two of these. Well, one of these can just go up this way and make this and then make this, and we have back to one again. So we still have our original oxaloacetate if I make an alpha-ketoglutarate from any of these nice structures. So those make sense. They can become a glucose. Same thing for this. This can become that second oxaloacetate, which becomes PEP, which becomes glucose. Same thing for this. And same thing if I just straight up make an oxaloacetate. I'm going to have two, and then one of them can just go to PEP. Why can't I do the same thing with acetyl-CoA? So again, we just have our one oxaloacetate. Why can't I just make an acetyl-CoA and then make an oxaloacetate and have all of that work the same way? Well, let's imagine now I make one of these. Well, acetyl-CoA is 2 carbons, oxaloacetate is 4 carbons, so they're going to combine. So now it's 0 and 0, 0 and 0, and I now have one of these. And then it reacts, and I have one of these, and it reacts, and I have one of these, and it reacts, and I have one of these, and it reacts, and I have one of these, and it reacts, and I have one of these, and it reacts, and I have one of these, and it reacts, and I have one of these. Can I go ahead and make a phosphoenolpyruvate from this? The answer is no, because I only have that one oxaloacetate. I need to keep it in the Krebs cycle. If it becomes a PEP, I now have no Krebs cycle intermediates, and I can't run the Krebs cycle anymore. So the point is that if you became acetyl-CoA and then you enter the Krebs cycle, you're using up the thing. If you go into the Krebs cycle only to then leave after making OAA to phosphoenolpyruvate, you're not really, you're using up your Krebs cycle intermediate. So hopefully that made sense. Play back if it didn't, because I think that's it's a nice connection. Otherwise, it's just rote memorization, and it's always good to make sense of things if you can. So hopefully that was clear. Glycogenesis and glycogenolysis. So I always hated the term glycogenesis because it really should be glycogen genesis, right? Glycogen genesis, the making of new glycogen. Because we have this here, like some douche pardon my French, thought it wouldn't be cool if we combine the gin from glycogen and genesis. And no, I actually think that makes it much more confusing. Here we have glycogen lysis. Glycogen lysis, we're breaking down glycogen. So making of new glycogen or the breaking down of glycogen. So all these beautiful alpha 1,4 bonds, and here we have this alpha 1,6 bond, the branching, and it's attached to this core protein glycogenin. So how do we make this? So glucose 6-phosphate is another really nice like intermediate. Glucose becomes glucose 6-phosphate. A lot of the fructose and galactose things can come in here. We can make pyruvate, also known as pyruvic acid, but we can also make glycogen, which is what we're going to be talking about. And later we'll be talking about the pentose phosphate pathway, also known as the HNP shunt. Taking glucose 6-phosphate and making glycogen genesis, also known as glycogenesis. Breaking down glycogen is glycogen lysis, glycogenolysis. And this glycogen is in liver, altruistic, muscle, selfish. So more specifically, glucose 6-phosphate via hexokinase or glucokinase if you're in the liver. We can isomerize it to become a 1-phosphate, boring. We can swap it with a UDP glucose, boring. Now is the more important stuff. UDP glucose can be, you can synthesize these alpha 1,4 bonds with glycogen synthase, and the branching enzyme can make these alpha 1,6 bonds. Again, remember it's like 1 in 10 of the bonds are going to be alpha 1,4 just to increase the density of the glycogen sugar. And when will we want to be building glycogen? Well, when we're in a fed state, when I just had a bunch of brownies and a burger and a milkshake. So insulin says, hey, we've got a lot of building materials. Let's build some glycogen. Let's store this away for a rainy day. So it's going to be building glycogen. Breaking down glycogen, so that's an anabolic process. A catabolic process breaking down then is glucagon. I haven't eaten in several hours and I'm really hungry. And so my body's like, hey, it's pretty important that we like feed the brain so that like he can, keep breathing. So it'll go ahead and tell the liver, can you break down some glycogen? Or you're, if you're running at the gym, you might need some extra thing to fuel in your muscles. So glucagon is going to help there as well. Epinephrine as well, though. Epinephrine is, maybe I'm not hungry, but maybe a bear is chasing me, and I don't know how long the bear is going to chase me. Maybe it's going to chase me for miles, right? So I would like to have a whole bunch of energy really quickly. The epinephrine can also facilitate the breakdown. The breakdown is going to be with glycogen phosphorylase is an alpha 1,4 breakdown, and the debranching enzyme is an alpha 1,6 breakdown. And breaking down, remember, is pyridoxal phosphate, which is a phosphorylase. This is a phosphorylase. We're breaking things down with phosphates, but its cofactor is the B6. And I just think of B6 as pure, as in Pyrenos. And I've said this a million times before. This is our altruist. It's pushing it's breaking everything down and put it out out of the cell into the blood to feed the rest of the body. And these guys are selfish. They get here and then they make the glucose not shown, and that just goes straight into glycolysis in the skeletal muscle. Pentose phosphate pathway. This is another really useful, cool intermediate. Glucose 6-phosphate. So glucose can become this, hexokinase or glucokinase. This can go to pyruvate, which is glycolysis, or it can go backwards as gluconeogenesis, glycogenesis, to make glycogen, or glycogen lysis to break it down. But we also now have the pentose phosphate pathway. And we don't, this is dash because we don't exactly go straight back to glucose 6-phosphate. It's a shunt because we're kind of like shunting it past, you're going back into the glycolytic pathway, but you're skipping a couple of steps. Hence the name is shunt. And you don't always do that either. So it's kind of an asterisk on that. The purpose, making NADPH, not to be confused with NADH. It's actually, it's a very similar molecule. It's just a phosphate on the 2' of the ribose structure of an NADPH. So it looks really similar, but it's crazy different. Making cholesterol, making fats, regenerating glutathione. You can also help macrophages create superoxides, hydrogen peroxides, and hypochlorous acid to kill things. And you can generate ribose, ribulose 5-phosphate, which becomes ribose 5-phosphate, which, DNA, deoxyribonucleic acid, RNA, ribonucleic acid. It's a ribose sugar in the nucleotide structure. So this is the precursor for that. So we have really two steps. The first half is going to be oxidative. This is irreversible, and the second step is non-oxidative, which is reversible. The most important thing is just knowing this first enzyme, glucose 6-phosphate dehydrogenase. It's the rate limiting step, the most highly regulated one for the pentose phosphate pathway. It's dehydrogenase, so we know we're making an NADH, an FADH2, or an NADPH, and here we're making an NADPH. We make more down the road, but this is the most important step, and if you know this, you're probably decent shape. We eventually can also make a ribose 5-phosphate, which we can see has two different decisions. We can go ahead and use that ribose sugar to make nucleotides, or we can convert it into fructose and feed it back into glycolysis. The choice is yours. So why do we call it the hexose monophosphate? I've kind of already answered this, is because we could go back into, we can shunt it around the glucose 6-phosphate and go back into glycolysis. So, we can go, hey, let's go here and let's make some nucleotides, or I kind of want to go back in, halfway through, maybe after having skipped a step or two into the glycolytic pathway, hence the name. And it's a hexose sugar, and it's a single phosphate on it as well. Cool. So glutathione. So the analogy kind of stinks here, but think of a reactive oxygen species as we're slow. We're passing the oxidation along. So this is getting oxidized to that, and then we can oxidize this into that, and then that can oxidize this thing, or it can oxidize other things. Or you're kind of passing the oxidation along, kind of like a hot potato. Now the analogy is not great because you're really not giving anything. You're actually taking something when you oxidize. You're taking electrons, but ignore that. And I just like to think of this as like a grenade, actually. So the reactive oxygen, we're passing oxidation from one molecule to the next, bing, bang, boom, bing, bang, boom, bing, bang, boom, passing along until you oxidize something that now it's really important. It was a protein. It was a nucleotide that we really wanted. And now this is the detonation, right? The detonation in the sense that it's permanently oxidized something which you're not going to reduce. You're now stuck with this hot potato. You're stuck with this oxidation, and it's a lipid. In our membrane, in our myelin, or it's a part of the DNA structure. It's part of the epigenetic regulation. It's a protein that we now can't have a hard time recycling, and that's why the reactive oxygen species are really bad within the cell. So glutathione is like heroically falling on this grenade. It is going to, it is reduced, and it is going to interact with hydrogen peroxide to reduce it. Putting it into the benign water. So. Yeah, it's jumping on that. Let's see that again. That's beautiful. Here's the grenade. It's going to damage something. No, glutathione jumps in and saves the day. So glutathione takes the bullet, jumps on the grenade, whatever you want, grabs the hot potato. But we need to regenerate it, right? We don't have an infinite supply of these. So we have the pentose phosphate pathway. So glucose 6-phosphate, the dehydrogenase here, this is the first step of PPP, can make an NADPH, which we saw. And it can pass this reductive potential onto a glutathione. So we mentioned earlier, this is kind of like a disulfide bond. This is a glutathione and that's a glutathione, and they are bonded together like this. Oil rig. Oxidation is lost, reduction is gain. We're talking about gain of electrons, which are formed in hydrides. This is a hydride. It's an electron-rich hydrogen. So going this way with this, we're giving the hydride over here. Now this has the reductive potential, which can sort of neutralize a dangerous peroxide. We've already seen this. This is the same thing again, right? The glutathione can pass it on to a vitamin C, which can do this, or the vitamin C can pass it on to a vitamin E, which I think could also do a peroxide, but often does lipids. Remember, the red blood cell membrane, we get hemolytic anemia if deficient, and you also are going to have the neuropathies because your myelin has tons of membranes which are being oxidized, and we want to reduce them. So, the NADPH, the glutathione, the vitamin C, the vitamin E. This is good too now. I wouldn't lose too much hair about I got to memorize every step of this, because I don't think you do. You often see, people, there's glutathione injections and creams you can put on, and a vitamin C and vitamin, not this one, that one, vitamin E. Ointments that you can put on, because really a lot of people think that aging or one of the factors, and probably the debate is okay, reactive oxygen species are bad in aged people, but to what degree? It's kind of like an apple. If you cut open an apple, this is really just oxidizing. It's oxidizing, and it's getting old really quickly, and that's a little bit what's happening to your face. So if we could cause a little bit more reduction, then maybe we'll keep somebody healthy and beautiful. Is at least the theory, and that's the rationale behind these ointments, creams, salves, medications. And again, the devil's in the details. Do they work and to what degree? Yeah, let's not get into it. So where does the pentose phosphate pathway occur? Red blood cells have a lot of oxygen. Oxygen can oxidize things. It can oxidize, you become superoxide, and hydrogen peroxide. And so, it's very combustible. So we have a lot of oxidation, so we need a lot of antioxidants. It's very delicate. In addition, it doesn't have like a nucleus and a lot of other structures to adapt and there's a lot of shear forces on it. So there's a lot of stress. And so long story short, you need a lot there. So they are going to have the pentose phosphate pathway. The adrenal cortex. Why? Because we are making a lot of cortisone, progesterone. Remember, all hormones that end in "-one" are nonpolar. Nonpolar hormones are made from cholesterol. Cholesterol is made from NADPH. The liver does make the cholesterol, so it also needs NADPH. Plus, the liver is, just as we said, involved in everything. Adipose tissue makes fats. If you make fats, NADPH will be helpful. And then lactating breast tissue. I guess there's a lot of fatty tissue in breast, and during lactation, there's more fatty tissue, or maybe you need more antioxidants for the breast milk. I don't really know, but it's also in lactating breast tissue as well. All right, moving on to carbohydrate metabolic regulation. There's a lot of different enzymes, and there's a lot of different things that inhibit all of those enzymes. And I think you are going to be very inefficient and drive yourself crazy if you try to remember, okay, this molecule for that enzyme and this molecule for that enzyme. If you just work from like first principles and have a nice foundation, this can be intuitive, it can be logical. So I think the first thing, even though I put it number 2, is negative feedback, right? If A makes B, which makes C, if I have a lot of C, it's going to feed back and inhibit A. This is going to happen all over the place. We've already seen this a lot. But then the glucagon versus insulin, which we're going to get into, glucagon really just works on like fat cells and liver cells. Its job is to say like, hey, we're really hungry. We need some more like resources in the blood. Can we like break down a lot of our stores? Can we liquidate our capital? And so I always thought of like, okay, in a time of hunger, you haven't eaten in a few days, who are the only cells that are really like high in ATP? It's going to be the wealthy cells. It's going to be the liver cells. It's going to be the fat cells. So those are the cells that are high in ATP. And they kind of ask for a donation. It's like, hey, like the rest of the body really needs some food. Do you mind donating like something? And then it'll, eventually trickle down or make its way to the hungry, kneecap cells and whatnot. So these ATP-rich cells, liver and the fat, they're going to donate a phosphate. And ultimately, that's going to somehow, bing, bang, bing, like make its way into a glucose. In the same way that if you had a big charity, it's like, okay, everybody donates $100 and I don't know, they end up building a school for some, lower socioeconomic neighborhood, or, vaccines and medications for like some developing country in need. And insulin is just going to be the opposite. Times are good. Everybody has exactly what they need. Nobody's hungry. So, keep your checkbooks at home. You don't have to donate anything. We'll get more into that. All right. This is glucose lysis. We're really just breaking down, oxidizing glucose. All cells, or most cells, almost always are doing this, right? So we can dial up a lot of these various processes that we're going to get into, but glycolysis is like, you kind of always need to be doing this. So I think of this as like, so glucose is like the money in your wallet. Glycolysis is like, I need to buy sandwiches to feed myself and I need to pay my mortgage. This is just money that I have to spend every day, no matter what, no matter whether times are good or times are bad. And when you're in the fed state, this is like, you're getting like, I don't know, you're getting like your payday, right? Every 2 weeks, you're getting a bunch of money from your work. And let's maybe you could go crazy, but maybe you're smart and you're like, okay, I'm going to invest this in stocks, or I'm going to buy like, a nice house or something. I'm going to put it in the bank. And then fasting is like, oh shit, all the bills just came in. We need to sell off some of our like stocks so that we can pay the tax collector. And then the various forms of capital where we're storing a lot of this stuff is going to be glycogen, fat, and protein, right? This is going to be, analogous to your stocks in your nice home and whatnot. So we always need to do glycolysis, like kind of no matter what. So that's not going to be dialed up or down too much in general. Gluconeogenesis, making new sugar, our liver does that, and it does that to feed the rest of the body when we are hungry. So that is only in the fasting state when our body needs sugar. Glycogenesis, making glycogen. This is only going to be in the fed state. You can only afford to store this away for a rainy day, push it in your special bank account of glycogen if you have just gotten a huge check in the mail from your work. Opposite is like, oh shit, there's so many bills that I have to pay. I need to take some money out of the bank. I need to sell my car. I need to somehow liquidate some of my capital to make money so that I can, pay my bills and feed my family. And so that's a fasting state. Lipid synthesis, fed state. Opposite, lipolysis and beta oxidation, fasting. And then ketogenesis, this one's kind of weird, but I would say this is in the fasting state where it's kind of like, remember that you can do gluconeogenesis, but you can also do ketogenesis. Some things like lysine and leucine can only be converted into ketones, which you can still use to fuel your body, and we'll talk about in a later lecture. Okay, so a lot of the regulation has to do with like never-ending debate between insulin and glucagon. Insulin is going to be the beta cells and glucagon is going to be the alpha cells, all from the pancreas. And really how they're working is they're putting on a phosphate and taking on a phosphate. That's really it. Insulin is typically taking off the phosphate while glucagon is putting on the phosphate. Insulin is really just doing the work of 4 different hormones. Insulin has to counterbalance epinephrine, cortisol, growth hormone, and glucagon, which are all, so this is going to be our anabolic, and these are all going to be our catabolic. So if we just ate a Pop-Tart, it's payday, we're a lot of cash monies. So we're gonna try to build stuff up. We're gonna go ahead and store this and invest in this big, production. I'm investing in a new company or something. And so what that means is we're going to be, making some glycogen. We're going to be making some lipids in the liver, but also in adipose tissue. And then, of course, the reverse is going to be turned off, right? If I'm making lipids, I don't want to be breaking them down. If I'm making glycogen, I don't want to be gluconeogenesis. Ignore that one. I was thinking glycogenolysis. I don't want to be doing glycogenolysis. Really, a lot of this should just be intuitive about what's going to happen. You can afford to build big fancy muscles if you're eating Pop-Tarts all day. I think that's how it works. And, so you're going to be building muscle, not breaking down muscle. Glucagon, so you're running from a bear, you haven't eaten in days, your blood sugar is pretty low. This is like, your bills are due. And so you need to take money out of storage, out of the bank, sell all your stocks so that you can get liquid cash right now, glucose to feed the hungry cells and whatnot. We need to pay our mortgage. And so this means we're going to be doing more gluconeogenesis. We're going to be breaking down fats and breaking down muscles and whatnot. So amino acid breakdown, and then some of those are glucogenic and then some of those are ketogenic. And so we might make ketone bodies at the end of the day as well. All right. G protein coupled receptor is going to be associated with glucagon. Receptor tyrosine kinase is going to be associated with insulin. So let's do a refresh on these. G protein coupled receptor is a 7-transmembrane alpha helix receptor. There's a heterotrimeric G protein when this whole thing is activated. So the glucagon binds, or epinephrine as well does the same thing. It activates the G alpha. It dissociates from G beta and G gamma. It goes over and is going to turn on adenyl cyclase. Adenyl cyclase turns ATP into cyclic AMP, which activates protein kinase A. It is a kinase. It is going to phosphorylate the bejesus out of everything else. And so you should think of glucagon as associated with lots of phosphorylation to regulate things. So this is a catabolic thing, is going to be associated with phosphates on everything. It's going to activate. So certain enzymes are going to be activated with the phosphate. Those are going to be the catabolic proteins. Certain enzymes are going to be turned off when they're phosphorylated. Those will be the anabolic proteins. Insulin is a receptor tyrosine kinase, not shown. We have these 2 receptor tyrosines that are binding insulin, and then they're dimerizing, and then they're phosphorylating their intracellular, tyrosine kinase domains. Blah 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 blah. PIP2, inositol. Remember, is this this phospholipid, and then inositol is the functional group that's attached on the intracellular side. PIP2, So we're going to have three phosphates basically on this, and phospholipase C. It's lipase. It's breaking the fat, and so these three basically PIP2 is IP3. The IP3 leaves from these two fats, so this just leaves behind a DAG, a diacylglycerol, really just like two fatty acids. And again, this is the inositol triphosphate, which is now left. That goes over to the smooth endoplasmic reticulum, which has a bunch of calcium sequestered on the inside. The IP3 binds. It lets out a lot of the calcium. Calcium is going to activate a bunch of different random things. And ultimately, though, what you should think about for insulin is we're going to be dephosphorylating. I know there's a kinase involved and you do phosphorylate some things, but the relevant metabolic enzymes will be dephosphorylated typically in response to insulin. So just, I kind of said this earlier, but if you're in tough economic times, who are the only people who have money? It's going to be maybe like the really wealthy people. And so we're going to ask them for donations. And that's because of glucagon. We're thinking about glucagon here when we're in a tough, fasting period. It's going to go to the liver and fat cells, the wealthy cells, the only ones with ATP. And we're going to say, hey, we really need you to donate. So that's why when we look at glucagon, we're going to see that we're phosphorylating everything. We're taking the ATP from the, this is a fat cell, or this is a liver cell, the wealthy ones. Wealthy doesn't mean bad in this analogy. Wealthy just means wealthy, right? They might be altruistic. So there's a lot of ATP in these cells, and the rest of the body is very hungry. And so we're going to ask for donations. And because we can afford it, we're going to phosphorylate all of our proteins. And this has a downstream effect of eventually generating glucose to push out into the bloodstream to feed the rest of the cells. I just thought of this. All right, so insulin is no phosphate on downstream enzymes. Glucagon is phosphate on downstream enzymes. So let's just look at an example. Glycogen synthase. This is building glycogen. That means it's anabolic. That means it's going to be stimulated by insulin. Remember that insulin is taking off phosphate. That's just what it does. You should just commit that to memory. And that's exactly what we see here. Insulin is empowering a phosphatase, which takes off a phosphate from glycogen synthase and therefore activates it. What if there's glucagon or epinephrine? Well, remember, they are going to phosphorylate the bejesus out of everything. And that is going to, they don't want this on, because why build glycogen? We need to break down glycogen to feed the rest of the body. So it is going to phosphorylate it, and that deactivates it. And the exact opposite can be said of the glycogen phosphorylase. Again, insulin still is taking off a phosphate, but this enzyme responds differently to phosphate. Here, when you take off a phosphate, it turns it off. And in this case, when you turn on a phosphate, or put on a phosphate, it turns it on. So this is just, it'll save you a lot if you can just remember this basic principle. This is how most of the enzymes work in terms of phosphorylation and dephosphorylation. So an example, protein X is catabolic. You eat a big meal. What happens? Insulin appears. What is insulin going to do? Insulin is going to take off the phosphate of a bunch of downstream things. Insulin also wants to turn on anabolic things and off catabolic things. So how do these things combine? We're dephosphorylating, and this is a catabolic protein that we want to turn on. What can we surmise about this catabolic X protein? That it is turned off by the insulin, dephosphorylation. That makes sense. And it's active when it is phosphorylated. What if we had an anabolic protein? Well, the opposite is going to be true. Insulin is going to dephosphorylate everything, and it's going to turn it on, and it's going to be made inactive by phosphorylation with like glucagon. Negative feedback, we've already talked about this, another regulatory principle. So we have a hydrocarbon like glucose, and we want to oxidize it to make ATP, NADH, and other things. Once we have enough of those, we go ahead and we feedback it until we say enough. Enough is enough and is enough. We worked really hard to make money, and we got enough money, so let's retire, at least for now, until we lose a lot of our money, and then we'll start over again. So we're going to see this show up. ATP, NADH is going to feedback at various points and say, enough, we're good. Stop working so hard. A couple examples. Glucose makes glucose. So it doesn't just have to be ATP and NAD. It could be the thing that's immediately downstream. It could be the thing that's a little bit more than a few molecules downstream. This feeds back, inhibits hexokinase, not glucokinase. Why? Glucokinase is in the liver, and in the liver we do not. It's not about. Well, you could have 5 brownies or 10 brownies or 20 brownies, right? There's no limit to how much sugar you can have, and we're trying to process this so that we can store it. So we don't actually want negative feedback in this case. Here we look at PFK1, enzyme number 3 of glycolysis. ATP is a downstream product. Citrate is downstream as well. So if you have high values of these, it tells you must have a relatively high energy building block abundant cell, which doesn't, can inhibit this. We can, slow the ropes with glycolysis. You can also think about like the energy state of a cell. If I have a lot of ATP, I don't need to do it. If I have a lot of ADP and a lot of AMP, that means I'm probably kind of cash poor, because if I was cash wealthy, I'd have more ATP and less ADP and AMP. So those are activated. On the other hand, AMP, which is, again, indicative of an energy poor state, is going to inhibit fructose bisphosphatase. If we look at pyruvate, similar thing over here. This is going to make acetyl-CoA, and then all of these things, the acetyl-CoA, NADH, ATP, are all going to negatively regulate the PDH, which is a highly regulated enzyme. More of the same. These are the two most important steps here, right? Remember, these are both taking off a CO2. So these are the two steps in a row that are taking off the CO2. Acetyl-CoA makes citrate. We isomerize it to isocitrate. That's boring. Now is the fun stuff. Pop off a CO2, make an NADH, make an ATP. But because these are the important enzymes, that they're highly regulated ones. And it should be, you should not have to memorize this at all. It should be intuitive. If you have a lot of ATP, we're good. Enough. We have enough NADH. Cut it out. This one's succinyl-CoA. Why? Because it's immediately downstream. Should you remember that specifically? I don't know if you should, right? Because are the questions really going to be, does succinyl-CoA negatively inhibit isocitrate dehydrogenase or alpha-ketoglutarate dehydrogenase? I don't think that's what the question will be. The question will be like, hey, what would happen to alpha-ketoglutarate dehydrogenase if you had a lot of succinyl-CoA? So I really don't think you have to get too granular. It should just make sense. So we just talked about this. The liver is a unique problem, right? Because for the liver, we actually don't want it to be able to, it's not like you can have enough end product, right? Because the end product is like storing glycogen, storing fat. And we might need a near infinite amount of that. Why? Well, because the opposite situation is just having a blood sugar of 1,000, which would kill you, right? So the liver has to detoxify the blood from sugar, and so there should be no limit to this. So what we did was, spoiler. So we showed that this is dealt by making it a glucokinase. What other regulatory hurdle might we encounter? Well, PFK1 is super highly regulated, and we need a way around this, just like we have the hexokinase to glucokinase switch. So really, to beat a dead horse, this is the problem, is normally this is how the inhibition works in a normal cell, but we do not want this in a liver cell. So what do? We switch it out for glucokinase, which is not inhibited by too much glucose 6-phosphate. But now we have another problem. And now the problem is that a bunch of things, high ATP, high NADH, high citrate, are going to go ahead and inhibit PFK1. I do not want PFK1 necessarily inhibited. I might want to make a bunch of acetyl-CoA so I can make fat in my liver if my blood sugar is too high. I don't want to inhibit this process. So what's the solution? The solution probably should be just similar to how we had hexokinase and we just make it glucokinase in the liver. What if we just had a different version of PFK1 in the liver? To me, that'd be brilliant, but no, the body's like more confusing than that. So instead, it has insulin override. So if my blood sugar is really high, I'm going to have a lot of insulin. Insulin overrides this negative inhibition from the negative feedback, and it's going to upregulate PFK1. But it's even more confusing than that, right? The devil's in the details. It works through a proxy. So, PFK1 makes fructose 1,6-bisphosphate, a similar molecule, fructose 2,6-bisphosphate. It sounds similar, but it's actually quite different. It is going to be the proxy through which insulin pulls the strings. It is going to be the switch which activates PFK1 and says, hey, I know there's a lot of ATP, I know there's a lot of NADH, but you can keep going. It's okay. The blood sugar's too high. Keep doing your thing. How does this work? Where does it come from? So fructose 2,6-bisphosphate is made by an enzyme PFK2, phosphofructose kinase 2. I think this is likely to show up in a question. One, it's just important, but two, Rem, I think, did a lot of research on this. I think if you look at the Wikipedia for this, you actually see Rem's name show up a couple times. Pretty cool. But this large enzyme, PFK2, actually has two domains. It is a kinase domain and it is a phosphatase domain. And the kinase domain is taking fructose 6-phosphate, the same, fructose 6-phosphate that becomes fructose 1,6-bisphosphate via PFK1 can also become fructose 2,6-bisphosphate via PFK2. it can be have the phosphate taken off to go from here to here with the phosphatase domain. The PFK2 kinase domain, if it's phosphorylated, it's off. If I take off the phosphate, it's turned on. On the other hand, the phosphatase domain over here, if it's phosphorylated, it's on. And if you take the phosphate off, it's turned off, which is very consistent with what we've seen with catabolic, anabolic effects of insulin and glucagon and phosphorylation. Okay, so just to tie everything together, the whole point of PFK2 is that if we have a lot of ATP, normally that negative feeds back on PFK1. But in the liver, if there's just a ton of sugar in the bloodstream, we want to deal with it. We want PFK1 to keep going. We need to override the negative feedback. So we want to turn it into pyruvate, which becomes acetyl-CoA into fat. How does this work? PFK is switched on, or there's more positive. Yeah, it's basically told to ignore the negative feedback. Switched on by fructose 2,6 bisphosphate, shown there. That is generated by PFK2, the kinase domain, and PFK2, the kinase domain, is remember that these regulatory enzymes typically, if it's this is going to be this is dephosphorylated by insulin, which is that's what insulin does. It dephosphorylates things. So insulin dephosphorylates PFK2. That turns off the phosphatase and turns on the kinase. The kinase turns fructose 6-phosphate into fructose 2,6 bisphosphate. That goes ahead and tells PFK1, hey, ignore all the haters, ignore the ATP, ignore the NADH, keep going because we need to deal with all of this sugar, turn it into fat to store it away. That's better than having high blood sugar. This is a more complicated picture, but I actually think you should really know this very well. I would know this inside and out. Hopefully this makes sense. Metabolic disorders. Okay, von Guerich's glucose 6-phosphate dehydrogenase, Pompe, McDardle. I think I squeezed another one in there, fructose and galactose and arsenic and mercury. So von Guerich is a glucose 6-phosphatase. Remember, this is the glucose 6-phosphatase highlighted. So what happens if we get rid of this? Well, this enzyme is really going to be present mostly in, maybe kidney a little bit, ignore that, mostly just liver. So the liver's job is it's altruistic, it's going to feed the rest of the body. You can't do that anymore. You can't do that. That is incredibly problematic. This is going to cause hypoglycemia. And you're now going to have, okay, what do we do with all the glucose in the liver? Let's just push it into glycogen. It causes the glycogen hepatomegaly to swell. We're also going to have more of this because, again, we can't push it backward into glucose. So we're going to have more pentose phosphate pathway. Okay, what are the effects of that? More pentose, what does this make? It makes NADPH, which makes fats. It also makes ribose phosphate, which makes nucleotides. If you have too many nucleotides, you can have too much uric acid from uracil, and this causes gout. So uricemia can be a thing, and you're peeing out the uric acid and cause gout, things like that. You're also making more NADPH, so you're also making more lipids, more cholesterol, more fats. Additionally, though, the reason you might have too much lipids is that because you're hypoglycemic, you have a high level of glucagon. The glucagon stimulates lipolysis, and adipose tissue breaks it down. So hyperlipidemia really just means there's a lot of too much lipids. Emia means blood, so it's too much lipids in the blood. So we're making too much lipids, and we're then glucagon's causing them to fatty acid mobilization, putting them into the blood, and so that's going to contribute to atherosclerosis and things like that. Just a nice little summary. Gout affects the toes, other places as well, but especially the toes. Hypoglycemia can cause seizures and intellectual disability. Glucose 6-phosphate dehydrogenase deficiency. I could be wrong. I think this is actually one of the most common metabolic hereditary disorders. So the dehydrogenase, remember, is going from this to this. And a dehydrogenase makes NADH, FADH2, or NADPH. That's the one it's doing here. It's the pentose phosphate pathway is making NADPH. So what happens if we get rid of this? We're not going to have any glutathione. There's a lot of different things that can happen. This is actually the only relevant thing. So the no glutathione makes the red blood cells especially vulnerable to reactive oxygen species. This leads to hemolytic anemia. This can cause jaundice. And it can be exacerbated by malarial meds, chloroquines, infections, flavivines. All of these lead to an oxidative environment. So that oxidation can damage the hemoglobin and cause it to precipitate. These precipitated damaged hemoglobins are known as Heinz bodies. When these red blood cells that have bad hemoglobin and are, kind of shitty, for lack of a better word, go through the spleen's job is to take them out of commission. Sometimes it doesn't do a perfect job. And the macrophages in the spleen come over and they try to gobble up the red blood cell, but it's elusive and it gets out of there, but not before the macrophage takes like a big old bite out of it. And so they call these bite cells too, if you see a red blood cell with a big chunk taken out of it. This, because it has a large role in red blood cells, has a protective effect against malaria. Malaria will be endemic to places in Africa and the Middle East and the Mediterranean. So look, if anybody, with that racial component or background, and that they are more likely, that might be a clue in the clinical vignette that they provide you in the NBME. And it's X-linked, so we're going to see it more in boys. Pompei's disease, so glycogen's broken down in a couple different ways and more complicated than we got into, but some of it gets shuttled into the lysosomes and muscle cells, and we have these alpha glucosidases, which help break it down. And they're defective in Pompei's disease. So we can't, our muscles aren't getting the energy that they need, and it's quite severe. You're going to see it it's called floppy baby syndrome sometimes, that the baby just doesn't have any muscle tone because its muscles aren't getting the energy that it needs. Cory's disease, you can't break down glycogen in the liver because so think about the debranch. So we talked about branching enzyme and debranching enzyme. Branching enzyme makes the alpha-1,6, debranching takes off the alpha-1,6. But it's actually, the debranching enzyme is a bit more complicated because, all right, we're chewing down these alpha-1,4, 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 alpha-1,4. Once we get to 1, 2, 3, 4, now the debranching enzyme comes into play. It has a glycosyl transferase, so it's going to take this and then, move it up there. And so now we see it right there. But it leaves this little guy behind. And to get rid of this, we're now... we now need to get rid of this. That's where the glucosidase comes in. And this glucosidase is not functioning properly in the Cory disease, so you can't properly break down glycogen. And so that your liver is it's hindered in its ability to fuel the rest of the body with sugar. Now, as opposed to glucose 6-phosphatase deficiency, von Guericke's disease, we can still do gluconeogenesis. So very bad disease, but it's going to be a little bit less severe than the glucose 6-phosphatase deficiency. You will still see these large, mainly hepatomegaly, these large livers, because there's so much glycogen there that we just can't break down. McArdle's disease, the muscle phosphorylase, so the alpha-1,4 isn't working in muscles. And if you can't break down, you're gonna have muscle pain, muscle weakness. The muscles are gonna be broken down, especially because you're gonna have red urine because you're breaking down muscles. The myoglobin gets released; it goes in the urine, red urine. It's because we're in such energy. Our muscles need so much energy, and a lot of that energy comes from the glycogen that they're selfishly hoarding. But if they can't access that, they're then going to require the liver to do extra gluconeogenesis, which is going to ironically kill the muscle cells that are needing the sugar in the first place. There's something called the second wind phenomena, where if somebody's exercising with McArdle's disease, they might have pain and fatigue in the first couple of minutes, but they get kind of a second wind where there's a little bit less pain and a little bit less fatigue after they've been exercising for a bit. I'm not really sure why. It's either they're switching their metabolic pathways, or there's vasodilation, which means that we're supplying those muscles with more nutrients, more sugar, and so they're not as starved as they previously were. I'm not actually sure. Poisoning. Arsenic and mercury can both inhibit lipoic acid, which is tender loving care for Nancy. L-loving, L-lipoic acid. So the PDH isn't working, and obviously that's not good. The Mad Hatter is, I think, people make like felt like caps. I don't know, back in like the 1800s, would use mercury to treat the wool, and I guess they would inhale the fumes, and it causes a lot of symptoms, neurological symptoms especially. Fun fact. Arsenic also robs us of an ATP during glycolysis. So it's shunting past the good part, right? If anything, we'd want to shunt past the investment phase. But no, it's shunting over the payoff phase. So, we spit glucose into 2,3 carbon structures. Now we're in the payoff phase, and we're making NADH and an ATP here, but nope. Arsenic's going to take this molecule and shuttle it all the way over here. It thinks it's helping, but it's not helping because we're not getting the NADH and the ATP that we wanted. So it's kind of like the 2,4-DMP shuttle, where it's moving things from point A to point B, but it's kind of missing the whole point, that we're not getting everything was set up in a specific way to get us various casino chips and ATP. So we talked a little bit earlier about galactose. So we can see fructose is turned into F1P with fructokinase, and that it can be converted into DHAP, which enters into glycolysis via fructoaldolase. So fructoaldolase is called hereditary fructose intolerance, and then fructokinase is essential. So we can block this one or block this one. These are two separate diseases. And this is similar to what we saw with the galactokinase and the galacto-1, And so which of these is worse and why? It's the second one. It would be worse to block this one and to block this one. Why? It's because if the fructokinase isn't working or the galactokinase isn't working, that's fine. The galactose and the fructose, respectively, they don't enter into the cells, because once you get phosphorylated, just like we saw with hexokinase, you're now trapped in the cell and you can't leave. So cells are just going to accumulate the fructose 1-phosphate and the galactose 1-phosphate and the fructokinase and galactokinase deficiencies, and that's bad. We'd rather pee them out or have them turned into something else. We don't want them trapped in the cell. It also wastes an ATP to phosphorylate these things. You're never going to get that ATP back. So we're also depleting some of our energy stores. So the first one is, if you're going to fail quickly, right? So we'd rather have the fructokinase and the galactokinase work. We're trapping them into the cells. What would the urine test say? Really just looking at this one specifically. If you have a fructokinase deficiency, you're going to have a lot of fructose still in the blood, and you're going to pee a fair amount of it out. So you might see a positive test for fructose in the blood. But if the fructose kinase is working fine, we're going to make fructose 1-phosphate, which trapped in the cell, and now it's trapped in the cell, which again, that's bad. That's a worse phenotype, and you're not going to see any fructose in the blood. Whew, okay, I know that was a lot. That was long. I talked fast. Hopefully at least made sense. So hopefully that was helpful. I hope everyone's doing well. All the best.